Are we live now?
Yeah, we are live.
Okay. Hello and Assalamu alaikum everyone, and welcome to the final session of the first annual Neuro-Oncology Symposium organized by the Pakistan Society of Neuro-Oncology. I, Rida Mitta, am your host for this session. We are honored to have Dr. Adnan Jabbar as a session chair with us today. Dr. Jabbar is Associate Professor and Section Head of Medical Oncology at the Aga Khan University Hospital. We have an esteemed group of speakers for today's discussion. Before we start, if any of you have questions for our speakers, you may ask on onlinequestions.org by entering the event number 13419. I would now like to invite our first speaker. Our first speaker is Dr. Tracy Batchelor from the USA, and he will be speaking on precision neuro-oncology, targeted therapies for brain tumors. Dr. Batchelor is a professor of neurology and neurologist-in-chief at Brigham and Women's Hospital, Boston, Massachusetts, USA. His primary field of clinical expertise relates to the therapy of malignancies of the central nervous system and neurologic complications. He also serves as the co-chair for the NCI Brain Malignancy Steering Committee and has held and holds numerous leadership positions within the American Academy of Neurology and the Society of Neuro-Oncology. Dr. Tracy.
Thank you very much. I want to thank the organizers for inviting me for this conference. It's an honor to be invited, and it's a pleasure to be with you this afternoon, in your case. So I'm going to share my screen here and pull up my presentation. Can everyone see this?
Yes, we can.
Okay, very good. Well, again, thank you very much. Pleasure to be here. And my topic this morning is precision neuro-oncology, targeted therapies for brain tumors. So this is very much a work in progress in terms of the introduction of targeted therapeutics to neuro-oncology, but we've had some initial successes, and I will predict that we're going to have more as we move into the future. So these are my disclosures. And this is a slide that is taken directly from the CTEP website of the National Cancer Institute. And this is a slide that highlights high priority cancer targets as deemed by CTEP or NCI for drug development. So many of these targets are highly relevant for brain tumors. It's a bewildering slide, but there's all sorts of, VEGF, for example, is on here, PDGFR, and others. So it's a very rich set of targets that are under development in cancer medicine. And fortunately, many of these will be relevant to neuro-oncology, as you will see, I hope, as we go forward. So I just want to start by mentioning that there are some substantial challenges that we face in neuro-oncology when it comes to single targeted therapeutics. These are a couple of papers that came out of our group here in Boston. This is a paper by Matthias Nudel. I'll refer you to the reference there for the details. But basically what Mattia did was he took autopsy specimens from glioblastoma patients at our hospital, and he analyzed different areas of these diffuse glioblastomas. And what he noted was that in different areas, using fish, for example, you could find different oncogenes that were amplified. For example, in this area, you might have PDGFR amplified, but in this area, you might have EGFR amplified. So this is known as genetic mosaicism, and I think it points out immediately a potential obstacle to thinking about using a single targeted agent like a PDGFR inhibitor or an EGFR inhibitor. So it turns out it's even more complex than that. So this is work by Anush Patel when he was a neurosurgical resident at our hospital in Boston. And Anush and his mentor, Mario Suva, looked at the transcriptional programs within single glioblastoma patients. And what they found, and there were 5 different glioblastoma patients here. These were autopsy specimens again. And they found that there were different transcriptional programs that were activated within the same tumor. So you might remember these transcriptional programs, these four classic categories: proneural, neural, classical, and mesenchymal. Well, it turns out that different parts of these tumors will have different transcriptional programs turned on. So there are definitely some challenges when thinking about targeted agents and single targeted agents. Nevertheless, there have been some success stories in gliomas. There are now 3 FDA-approved targeted therapies for neuro-oncology indications, and I predict that there soon will be more. I'm going to just walk through each of these very quickly. There's everolimus, larotrectinib, and selumetinib. So everolimus is an mTOR inhibitor, and it is approved for the use in tuberous sclerosis patients who have subependymal giant cell astrocytomas. So TS is an autosomal dominant genetic disorder. It's caused by deregulated mTOR signaling. About 20% of TS patients develop these subependymal giant cell astrocytomas. As you can see here, they tend to have these, again, subependymal locations. I mentioned that everolimus is an mTOR inhibitor, and this was a trial of everolimus in patients with TS who had SAGAs, subependymal giant cell astrocytomas. And so they looked at a the 5-year point, they looked at a number of outcomes, but their primary endpoint was the volume of the tumor. And in these patients, the median duration of treatment had been about 5 years on the drug. So even though there are some side effects of this drug, it can be given over a long period of time. And what they observed was that over half of the patients had reductions in their tumor volume by more than 50%. And you can see some of these cases here. These were not necessarily complete responses, but definitely partial responses. And also another endpoint of this study was the number of patients having daily seizures. So this is, as you probably know, a major complication of TS are seizures. And you can see that in the patients who received the everolimus, the 26% were having daily seizures at baseline, and that went down to 11% after treatment. And importantly, no patient had to discontinue this oral agent due to adverse effects. So the FDA granted approval of everolimus for subependymal giant cell astrocytomas in patients with tuberous sclerosis in 2010. Selumetinib is a more recent success story. So this is an oral MAP kinase inhibitor, and this was studied recently in neurofibromatosis type 1 patients who had progressive plexiform neurofibromas. These can be disabling tumors. They can cause disfigurement. They can cause pain, disability, and there's not really any effective treatment for them. However, they seem to be driven by increased signaling in certain MAP kinases. And so selumetinib was studied in patients with NF1 who were 2 years of older, who had symptomatic, inoperable plexiform neurofibromas. And so they ran initially co-clinical trials in mice models of this tumor and in humans. And in the phase 1 human trial, 24 subjects, the median age at diagnosis was 10, 70% of patients had partial responses. No patients had disease progression while on the drug, and there were anecdotal decreases in tumor-related pain, disfigurement, and functional impairment. So based on this study, this phase 1 study, and a follow-up phase 2 study, the FDA recently granted approval of selumetinib for the treatment of NF1-related plexiform neurofibromas. Here you can see an example of a case with a diffuse plexiform neurofibromas causing disfigurement, and you can see over time the selumetinib with a partial response in this patient with improvement. And here are the waterfall plots at different doses showing that practically all cases had some level of tumor response. And finally, I want to mention larotrectinib. And before I talk about larotrectinib's approval by the FDA, I'll mention some work that we did at our institution a few years ago when we looked at a collection of about 25 glioneuronal tumors, and we looked at different genetic alterations in these tumors. And we observed that in about 1/3 of these 26 tumors, there was a fusion with an oncogenic fusion with NTRK. So fusions involving the NTRK family have been reported in other cancers. They constitutively activate TRK protein kinase signal transduction, and they're felt to be oncogenic events. So again, about 1/3 of these glioneuronal tumors had oncogenic NTRK fusions. And we had, at the time, a phase 1 trial of an NTRK inhibitor, in this case, entrectinib, so that it was disease agnostic. So any solid tumor that had an NTRK fusion could be enrolled. And this is an example of a glioneuronal tumor that we enrolled on this study. This is prior to treatment with entrectinib. And then after treatment with entrectinib, you can see a significant partial response, which was maintained for about 4 to 6 months. And unfortunately, in this case, the tumor progressed. The NCI ran a large trial of NTRK altered solid tumors, looking at the drug larotrectinib, which is another NTRK inhibitor. And based on outcomes and responses in that trial at NCI, larotrectinib was granted FDA approval last year for any solid tumor that has an NTRK fusion. So it is available to our patients if they have that particular genetic alteration. So those are some success stories that have led to FDA approval. These drugs are available to us to use in these specific indications. But now let's talk about what's in the pipeline. So staying on this theme of pediatric tumors, I want to talk a little bit about pediatric low-grade astrocytomas. So these are characterized by alterations in BRAF in about 75% of pediatric low-grade astrocytomas. They're the most common oncogenic driver of this tumor. However, the majority of these oncoproteins are not point mutations like the V600E that you see in melanoma and other tumors, but they are actually a truncation fusion variant. That's the majority. And it turns out that the first generation BRAF inhibitors do not work in patients who have these truncation fusion events. So they're really ineffective, and it was not really known why these did not work in these truncation fusion events. But we have done some studies of utilizing our medicinal chemistry core and a structural biologist on a grant that we have called a SPORE grant, where we identified why these truncation fusion events are resistant to these type 1 inhibitors. But we've also identified a host of type 2 RAF inhibitors that do work well. And this was, I don't pretend to understand the crystallography here, but I think it points out the importance of incorporating correlative studies and looking at some of the structure-activity relationships to better understand how these drugs work or don't work. And so Mike Eck, who is a structural biologist in our program, published this letter to Nature last year demonstrating this BRAF MEK complex and why these type 2 RAF inhibitors actually work. And this is an example of a patient treated with a type 2 RAF inhibitor, a pediatric low-grade astrocytoma, showing a reduction in size of the tumor, and another one here. So these drugs are now in clinical trials, and we will see where they end up, but they're certainly showing promise. So in order to do these types of studies, one has to have a robust molecular profiling platform. And this is the system that we use at our hospital, where we do the basic immunohistochemistry and histopathology, of course, within a few days. Then, of course, with certain types of tumors, the MGMT status is very important. So we tend to get this a few days later, but usually within the first week, so we can make a decision if there's a trial revolving around either methylated or unmethylated cases. And then we have a SNP array oncoscan that'll pick up about 800 genes, cancer-related genes that is available within 2 weeks. And then we can do either targeted exome sequencing or even whole exome sequencing as the case may require. This requires a whole integrated team of pathologists and clinicians, and the readout has to be and readout with an integrated diagnosis into the electronic medical record, which would include all of this. But this then enables us, to think about, again, if there's an NTRK fusion or something like that, we may have an FDA-approved drug to use. But more importantly, I think right now is a lot of the trials that we are conducting are trials of targeted therapies, and we have to know that the target is present in the tumor. So this enables us to do that and enroll patients on clinical trials. So with all that being said, let me then switch and talk a little bit about some specific targets in adult gliomas, and we'll start with the epidermal growth factor receptor. Excuse me. As you know, that there are about 40% of glioblastomas will have amplification of EGFR, some cases high levels of amplification. And about half of those, or about 20% of patients with glioblastoma, will have an EGFR V3 mutation. Now, unfortunately, even though this is a prevalent target and is a known oncogenic driver in other solid tumors, the first generation EGFR inhibitors, gefitinib, erlotinib, et cetera, have not been effective for EGFR, for glioblastoma or glioma. So, but that's not to say that we've given up on EGFR inhibitors. So this is a study that we have recently published in the Journal of JCO Precision Oncology of an irreversible CNS penetrating EGFR tyrosine kinase inhibitor. And our eligibility in order to get in this trial is that you had to have EGFR amplification or a mutation. So these were recurrent glioblastoma patients. We determined whether they had an EGFR event or not, and you had to have one to enroll in the trial. And by the way, this trial was open also at Henry Ford Hospital in Detroit, with Tom Mickelson was there. And then we stratified by prior use of bevacizumab, and then we had a surgical arm so that you were eligible for resection. We would give you the drug, this EGFR inhibitor, dacomitinib, for about 7 to 9 days. We would operate, and then we would look for presence of the drug and some evidence of drug activity in the tumor. Then there was a non-surgical arm. This was the main arm of the trial, where we looked at a phase 2 efficacy endpoint here. And then there was a third arm in patients who had had prior bevacizumab. And as you know, these are a particularly poor prognosis group of glioblastoma patients. So here's an example of a response to dacomitinib in the trial. Most patients did not respond. There were a handful, and the progression-free survival at 6 months was about 25%. So it was not what we were hoping for with this drug in this EGFR amplified group of patients. But we also incorporated some correlative studies into this trial, and one was to look at exosomes and the content of exosomes to determine if we could pick up a reason why these tumors might be resistant. So what are exosomes? Exosomes are circulating membrane-bound vesicles. They basically emerge from the intracellular compartment where they form on the cell membrane, and then they eventually pinch off and they circulate. And when they pinch off here and form this vesicle, they will capture some intracellular content, including RNA. So you can actually do targeted sequencing of the RNA, and you can look at certain genes that are activated in this tumor that are actually circulating in exosomes. So we did this in this trial of dacomitinib, and we divided our population into 2 groups: those that were rapid progressors within 3 months of treatment, and those that were responders. That is to say, they had no progression 6 months or later. And you can get a qualitative sense from the RNA in here that there are differences in the RNA content between these rapid progressors and responders, and that we do have a lead on a couple of genes in the rapid progressors that may be resistance genes. And so this is very, very early work. We have to come back to it. But I think, again, this is a very simple, you just collect blood on these patients. So by incorporating this blood test, essentially, you are able then to maybe do a more refined analysis as to why some patients may have done well and why some patients may have been resistant. And I'm going to switch gears now and talk about another target, which is a very topical these days, which is IDH1, isocitrate dehydrogenase 1. So this is a gene that is mutated in about 10% of glioblastomas, but in about 60 to 70% of anaplastic and low-grade gliomas. This is just from a paper in the Archives of Neurology, pointing out how this is truly a diffuse glioma. So IDH1 mutant glioma, and all of the staining dots here are IDH cells. You can see that these gliomas tend to be quite diffuse, not only where the mass is, of course, but even into infiltrating brain on the ipsilateral and contralateral side. I think pointing out that the solution to this diffuse glioma really does have to be a systemic one, a systemic type of treatment or approach. And this is just a technique that I'll come back to in a few minutes, which is looking at IDH mutant gliomas by accumulation of an oncometabolite that is formed 2-hydroxyglutarate. You can actually measure that using spectroscopy. And this is just an interesting image that it shows the red outline showing the outline of the T2 flare tumor, and then the blue-yellow map is spectroscopy showing is the 2HG map, showing that the IDH mutant cells are much more diffuse than what we can even see on the T2 flare image. So this is a gain-of-function mutation, IDH1. Again, it leads to the accumulation of this oncometabolite, 2-hydroxyglutarate, which fortunately can be measured effectively with immunohistochemistry, 90 to 95% effective in picking up IDH1 mutations. Of course, when this is negative, you have to resort to sequencing, but it's something that can be easily measured, that is to say, an IDH1 mutation. So this is the technology that I mentioned to you earlier that has been developed by Ovidio Andronisi, who is a spectroscopist at our institution. This has also been developed by other institutions as well. But basically, it's a non-invasive MR way to measure 2-hydroxyglutarate in the brain and to pick up IDH1 mutations. So here you can see this is an anaplastic astrocytoma that has an IDH1 mutation. Here's the tumor, here's the voxel, and you can see in that voxel there's a 2HG signal indicating that this is an IDH1 mutant tumor, whereas here is a glioblastoma. Turns out it's wild type IDH1. Here's the voxel, and where you should see 2HG, you don't see it. And of course, here's a healthy volunteer, wild type, of course, and you can see there's no signal. So 2HG is elevated dramatically in these IDH1 mutant tumor cells, which enables it to be measured with spectroscopy. So this is an example of incorporation of this tool, if you will, this MR spectroscopy in a clinical trial. So the first generation IDH1 inhibitors that have been studied for IDH mutant gliomas have not really been as successful as these inhibitors have been in IDH1 or IDH2 mutant leukemias. There's been a lot of speculation as to why that may be. But this was a study that we were asking the question, this is an IDH1 inhibitor trial, and we are performing spectroscopy before and after administration of the drug. The red curves are the 2HG levels before treatment with an IDH1 inhibitor. The blue curves are the 2HG levels after treatment. So in this case, you can see that even though this was a negative trial, the drug did not meet its endpoint in this trial. It's not because the drug was not reaching the target. The drug is reaching the target and modulating the target because the 2HG levels are dropping in each of these cases. So this again is an example of incorporating a tool like exosomes that can help us understand why these tumors might be refractory or resistant. In this case, demonstrating that it's not an issue of drug delivery. The drug clearly is getting there and having some biological effect on the tumor. But as I mentioned, these IDH1 inhibitors do not seem to be that active in these tumors. And there are a number of potential reasons for that I won't go into. But we have adopted a different strategy in these tumors, and that is to look at the concept of synthetic lethality. We and others have identified that the mutation and the accumulation of the oncometabolite 2-HG creates a number of metabolic vulnerabilities of these IDH1 mutant gliomas. And we can use targeted drugs to capitalize on those vulnerabilities. And this is, I'm going to show you one example. There are several different strategies, but this is an example that involves NAD metabolism. So again, these are IDH1 mutant gliomas. They have accumulation of 2-HG, and normally in a normal cell, NAD is the product of a number of different pathways. Two key pathways are through nicotinic acid and this enzyme NAPRT1. The other pathway is through nicotinamide through this enzyme NAMPT or NAMPT. So this is how NAD is biosynthesized. And of course, we know that NAD is essential for cell survival. So what happens in an IDH1 mutant scenario is that there is accumulation of 2-hydroxyglutarate, and the 2-hydroxyglutarate turns out blocks one of these two pathways, this NAPRT-mediated pathway. That then renders this IDH1 mutant cell particularly vulnerable because it's now reliant on one key pathway for NAD biosynthesis. And so by taking advantage of this genetic lesion, we can then use a NAMPT inhibitor and hit this pathway and shut down NAD biosynthesis selectively in IDH1 mutant cells. And you can see from a series of experiments, this is exactly what happens. If you look at cell lines, all these black cell lines are IDH1 wild type, and the colored cell lines are IDH1 mutant. So when you use a NAMPT inhibitor, you selectively kill these IDH1 mutant cells. You can see also the tumor volume decreases in a model of IDH1 mutant glioma. The Ki-67 proliferation index drops when using this NAMPT inhibitor, and the animals live longer. So this is treated with a control, and here are animals treated with a NAMPT inhibitor. So NAMPT inhibitors have some promise in IDH1 mutant gliomas. There are NAMPT inhibitors under development. The first generation inhibitors really did not make it out into clinical trials because of some toxicities, but the second generation inhibitors, there are second generation inhibitors under development, and we hope to study one or more of them in IDH1 mutant gliomas. So in the last part of the talk, I want to just mention a couple of other brain tumors. So we're switching gears away from gliomas now to the most common primary brain tumor, which is meningioma. Accounts for about 1/3 of all primary brain tumors. And this was work done by my colleague, Priscilla Brastianos, and her team, where they conducted whole exome sequencing of a series of meningiomas a few years ago, and they identified some potential oncogenic mutations that could be targeted. So these are now, the most common genetic event, of course, is NF2 loss in meningiomas. Up to 50% have NF2 loss. So that's a well-known genetic alteration. So what this team did was they looked at the NF2 wild type samples. So these are meningiomas that have NF2 intact, and they then did whole exome sequencing on those, and they found that about 10% of these NF2 wild type meningiomas have an oncogenic mutation in AKT1, which had been previously described and is known to activate PI3 kinase signaling. So about 10% of NF2 wild-type meningiomas, AKT1 mutation. About 5%, again, in these NF2 wild types, had smoothened mutations. So another oncogenic driver activates the sonic hedgehog pathway. And of course, there are drugs available that target both AKT and smoothened. So this then, the other interesting thing about this is it seemed to be, there seemed to be some relationship of these AKT and smoothened mutations to anatomy of the meningiomas. 26 out of 38 of the skull base meningiomas had AKT mutations. 7 out of 11 of the skull base meningiomas had smoothened mutations. So that is a clue then in terms of the imaging that there might be some meningiomas more susceptible to these mutations than others. And as the surgeons on the call know, these can be the more challenging surgical cases can be these skull base tumors. So if we had drugs that might be effective for this group of tumors, it would be a positive development. And now this is, that particular work has been duplicated, and the group at Yale published their paper about the same time, also showing smoothened and AKT mutations. And now there's a whole host of genomic studies and meningioma identifying other targets. So the era of targeted therapies has started for meningiomas. This is an alliance study that is ongoing, and it basically is a study of targeted therapies for molecular subtypes of meningioma. So meningiomas that have a smoothened mutation would be treated on vismodegib, smoothened inhibitor. Meningiomas with an AKT mutation would be treated with an AKT inhibitor. And there is an arm for those patients who also have NF2 loss. There's some evidence that FAK inhibitors can be effective in the setting of NF2 loss. And in fact, this arm was just reported at ASCO as a plenary session a couple of months ago. This FAK inhibitor was studied in progressive meningiomas with NF2 mutations. And this study, phase 2 study, not a randomized study, but a phase 2 study, it met its primary endpoint of progression-free survival at 6 months. So this will hopefully move ahead, and we'll see maybe this drug may be active and available at some point for NF2 altered progressive meningioma. And let me end with another tumor. We go from the most common primary brain tumor to a rare intracranial tumor, craniopharyngioma, well known to our neurosurgical colleagues. This is again work by Brastianos and her team, where they looked at exome sequencing in craniopharyngiomas, and they identified in the papillary craniopharyngioma subset that nearly all of those cases, 95% of papillary craniopharyngiomas, have BRAF mutations. And in this case, these are the canonical V600E activating point mutations. And based on that, we then had the opportunity to treat a patient with a combination of a BRAF inhibitor and a MEK inhibitor. This is a 38-year-old recurrent papillary craniopharyngioma. Had multiple attempts at resection, as you can see here. This tumor by immunohistochemistry was BRAF V600E positive, so there's a BRAF mutation. And so after all of these decompressive attempts, the patient was eventually treated, first with a BRAF inhibitor alone, and then with a combination of a BRAF MEK inhibitor. And you can see a significant reduction in the size of the tumor. And at this point, the patient underwent resection. Here's just an example of a plot showing the volume of the tumor over time on the combination of the BRAF MEK inhibitor. So here is the pre-treatment surgical specimen showing the features of a craniopharyngioma, and then post-treatment, after treatment with a BRAF MEK inhibitor, you can see a lot of fibrosis and a lot of infiltration into the tumor by foamy macrophages, indicating some treatment effect. Similarly, you can see a drop in the proliferation index, influx of macrophages, et cetera, showing a real a biological effect of the drug on the tumor target. So that's a brief summary of and a snapshot of some of the areas that are under active investigation. By necessity, it was just a snapshot. There are many other targeted therapeutic approaches ongoing for glioblastoma or gliomas. We didn't even touch on brain metastases, where there's also a lot of work. So I would say it's a very exciting time right now in terms of the study of targeted therapeutics in neuro-oncology. And just a few principles that I'll leave you with in terms of target selection. We want to know, if we can, that it is a driver mutation, that we can actually measure the target in the tumor before we give the drug, so that we can select patients for the trial. You know, we want to choose our drugs in the neuro-oncology business wisely. They need to be very potent inhibitors of the target, and they need to be CNS penetrant, and hopefully not a substrate for drug efflux pumps from out of the CNS. Measuring the target is important. So fresh tissue is always best if you can do that. Use of a validated genotyping platform, that's very important when looking at clinical trials, especially multi-center clinical trials. Can we measure the drug in the tumor? Does the drug get into the tumor? Does it engage the target? And does it modulate the target? And I've shown you some examples of how we might be able to do that with tissue, imaging, and circulating biomarkers. And then, of course, understanding refractoriness or resistance to these drugs. And that's where it's important to have pre and post tissue, and also the circulating biomarkers might be of interest there, or of help there. And then, of course, we want to determine benefit, and that's where we get into the more classic trial endpoints like response, progression, survival, and then, of course, other patient-reported endpoints as well. So with that, I will thank you for your attention, and I will turn it back over to the organizers.
Thank you, Dr. Batchelor. That was just amazing how you were able to put all that into one small half-hour talk. Sorry, off to Dr. Jabbar, the chair of the session.
No, thank you so much. It's really intriguing time for us when we see precision medicine. You were right about, we haven't even gone into the metastatic lung cancers with alectinib, but really fascinating data. And thank you very much for explaining it very clearly and in very precise manner. We'll discuss more during our Q&A session. I would ask Nidha to go ahead and introduce our next speaker.
Second speaker is Dr. Jalare Zadeh from Canada, and she will be speaking on clinical impact of integrating methylation into management of brain tumors. Dr. Zadeh is professor and chair of neurosurgery at University of Toronto, head of division of neurosurgery at Toronto Western Hospital, and senior scientist at Princess Margaret Research Institute. On September 1st, she stepped into the role of the Dan Family Chair of Neurosurgery at the University of Toronto, becoming the first woman to helm one of the largest neurosurgical programs in the world and the first female neurosurgery chair in Canada. Dr. Zadeh.
And she is also going to be, sorry, Larry, and she's also going to be the president of SNO. I have to mention that. I missed that in the write-up.
Thank you, Professor Enam, and thank you, Rita. I'm going to share my screen now. You can see my screen?
Yes, we can.
Okay. I just wanted to first start off by congratulating Professor Enam and the committee for putting together this first annual Neuro-Oncology Symposium, and really for the tenacity to bring together so many endorsements from a number of different societies of neuro-oncology across the world. And it's been wonderful to see that you have so many esteemed speakers from across the world. Many of my colleagues, in particular, I'm glad to have been able to see Tracy. I haven't seen him for a long time, but more importantly, local speakers as well. And so I think the program has been very impressive, and hope that the momentum of this can continue, and happy to see how as SNO we can partner with you to make that happen. So I wanted to speak about the use of methylation and integration into clinical practice, and it really follows very nicely from Dr. Batchelor's presentation. And this is really to look at how can we improve on brain tumor classification. If you look at the WHO classification, there's over 150 distinct brain tumor types. Of course, on a day-to-day practice, we don't deal with 150 on a regular basis, but a considerable number of them. And there's always a challenge in diagnostic accuracy, and sometimes a couple of expert neuropathologists are unable to reach consensus on the diagnosis of particularly challenging cases. And if you look at, again, what Tracy presented and the history and evolution of how we understand brain tumors, we've moved away from the traditional histology to some extent and incorporated molecular features such as IDH status, 1p19q, ATRX, p53, to further define and refine our diagnosis. In particular, this bears on outcome for the patients and how we counsel them, how we decide on treatments, and then hopefully in time, the molecular knowledge that we've gained will help design the targeted therapeutics that we heard in the past talk. In 2018, approximately 2 years ago, there was a very seminal publication in Nature by the group in DKFZ, really overseen by Andreas von Daimling as the head of neuropathology at that department, where they used DNA methylation on 82 different tumor types. And as you could see, the methylation pattern of these tumor types is very distinct. And so the IDH mutant tumors fall into this category, whereas the GBM IDH wild type fall into this section of the methylation. And then, as you can see, there's a number of different subtypes that you could see of the gliomas, but also other tumor types that you have within the WHO classification that I showed you. And so really showing to us that methylation can be used to refine and add and complement the histopathological diagnosis that we make on day-to-day practice for neuropathology. So at our center, recognizing the value of methylation, we wanted to see what the clinical utility of methylation was and what percentage of cases that were challenging would actually have a change in diagnosis. And more importantly, of those, what percentage actually would have an altered clinical management. We obtained ethics approval and did this retrospectively on the cases that had methylation. And we looked at our cases between 2015, which is where, when in Toronto we established methylation as a test, up to 2018 is the course of the study, recognizing that we've continued to do the methylation because of its impact. And in this period, we reviewed 1,670 cases where we had tissue and complete clinical history on. And the clinical challenging cases were defined where 2 experienced neuropathologists were unable to reach consensus on the diagnosis, or cases where there was a discrepancy between what we felt as clinicians, particularly neurosurgeons and neuro-oncologists, imaging factors, and what the histopathology indicated, and in cases where we needed more molecular subtyping. So for example, of all the molecular targets that Dr. Batchelor was mentioning in the prior talk, methylation can in fact give you majority of that information. Using this inclusion criteria, of the 1,700 approximate cases, we identified 55 challenging cases to be reviewed. So for the methylation study, really just to put it briefly, what you need is to get DNA extracted from the tumor, and this is fairly simple. It's a DNA extraction kit that we use in any laboratory. We need paraffin blocks that are cut, 5 cuts that are unstained. We can extract the DNA, run the methylation on the Illumina array, and then be able to upload the IDAT files that are obtained on an internet-based DKFZ website that would then give you a score and a diagnosis, and then a readout of what the tumor methylation status would be, and complement your histopathology with the diagnosis given by methylation. So our workflow was, as you could see here, that we use the methylation-based classification. We sometimes use copy number to refine that classification. We incorporate the histopathology that the pathologist has given us, and consider some of the standard molecular tests that are done on routine fashion. For example, for 1p19q, we use FISH at our center to reach a final integrated diagnosis. This integrated diagnosis went back to the tumor board for a discussion, and you could see that the tumor board is consistent of the standard membership for the tumor board. So this is some of the data on this cohort. The mean age is 41. The gender is approximately 50-50 for male-female distribution. Majority of the tumors, as expected, are supratentorial. Skull-based tumors often are not a conundrum for diagnostic purposes. And the status of the patient was documented. The initial grading that was given by histopathology was documented as well, and subsequently the corrected grading of the tumor based on the methylation was documented. The tumor types, as you can see, majority of them are diffuse gliomas, IDH mutant, IDH wild type, and pilocytic astrocytomas, ependymomas, and some neuronal tumors, meningioma, and metastases. So this is the table that demonstrates what are the impacts of the diagnosis. 13 cases, 24% had a change in diagnosis. 31% had a resolution of a differential diagnosis, where again on histopathology, the pathologists were unable to reach consensus. Molecular features of the tumor were refined in 29% of the cases. And most importantly, an upgrade in the WHO grade of the tumor was made in 18% and a downgrade in 5% of the cases. And in 2 of the cases where a grade could not be assigned, methylation allowed us to do that in 4% of the cases. So this diagram shows you the initial pathological diagnosis that was made based on histopathology and molecular testing that's routinely done in the pathology lab. And this column shows what the final integrated diagnosis was. And as you can see, there is a considerable amount of crossover from the initial to the final integrated diagnosis. And as I showed you in the percentages, it's a considerable change in diagnosis. And so when we took back the final integrated diagnosis using methylation to our tumor board, the question is, did this in fact change management for our patients? And this is an example. I'll quickly go through this case for interest of time. I'm happy to come back to it, where the diagnosis was changed dramatically and the patient's treatment plan changed significantly as well. And so if you took all the cases that we had, the noteworthy ones are, for example, where an initial diagnosis in a 20-year-old lady who presented with seizures was glioma not otherwise specified, where the IDH was identified to be negative, and the tumor on histology did have what was concerning for high-grade features. The tumor board had reached a decision to give temozolomide and radiation. However, following the integrated diagnosis using methylation, the diagnosis was reached to be a DNET. And of course, in this scenario, resection was very nice, observation was recommended, and the patient avoided chemoradiation, which would be an impact in a positive direction for this patient. The other cases are a similar representation of where radiation and its side effects are avoided, given the fact that the diagnosis was changed. And in a few instances where a BRAF inhibitor, for example, would be beneficial, was considered, whereas in its original diagnosis, it was not. So with this, in summary, 84% of the diagnostically challenging cases received a clinically meaningful diagnostic alteration. 24% had a change in diagnostic entity, 31% had a resolution of their differential diagnosis, and 27% had a change in tumor grade. And ultimately, this change in histopathological grading and diagnosis resulted in 15% significant change in clinical management, which is a considerable and noteworthy percentage. And this work was published in Clinical Epigenetics. One of my students, Jeff Zuccato, together with a postdoc, Shireen Karimi, put the data together to demonstrate to our institution the value of methylation, to also be able to demonstrate to our ministry for funding purposes that 15% is a significant impact on the patient's outcome, and this test should be funded within our institution. And we're able to continue this program locally, but also serve as a referral center for many sites across the world where the pathology is uncertain, and we help with methylation to resolve the diagnosis. I do want to give special credit to Ken Aldape, who introduced the methylation at our institution when he was here with us, and thereafter, given its impact, we've continued with this program. I also want to take a few minutes to put a plug in for the SNO annual meeting. As Professor Anna mentioned, I'm the President of Society of Neuro-Oncology, an unprecedented, unpredictable period to be the President of SNO. It's our 25th anniversary. The meeting was to be held in person in Austin, but we're now moving to a virtual conference, which has its positives in that we can have more far-reaching impact, and I hope that many of you who are able to join us can participate in the meeting. And I also want to mention, again in discussions with Professor Enam, highlight the opportunity for the international scholarship. It's a scholarship that I introduced in 2015 with our local institution at University of Toronto, the Wilkins Barrick family, and SNO in partnership to offer scholarships internationally to help symposiums such as this. And I really hope that you will consider applying in the years to come so that the momentum can be sustained for what you've built here today. Thank you again for your attention, and I think you said that we take questions at the end. Thank you.
Thank you so much, Professor Zadeh. It was an excellent talk. A 15% clinical impact is significant, and I think this is something that we are seeing more and more, that impact of molecular testing on neuro-oncology. I must say that I've attended your SNO meetings, and in fact, I am attending your current webinars that you are doing. So I'm a current visitor of that, and I'll certainly spread the word regarding the opportunity for our fellows who are interested in the field. We'll take further questions later on. Rida, please introduce the next speaker.
Our third speaker is Dr. So-Gu Kang from Korea, speaking on a translation niche with glioblastoma origin cells from mouse to human subventricular zone. Dr. Kang is Professor of Neurosurgery at Yonsei University College of Medicine, Seoul, Korea, and the Vice Chair of the World Federation of Neuro-Oncology Societies, WFNO, Scientific Committee. His field of interest is surgical neuro-oncology and translational research in brain tumors. Over to you, Dr. Kang.
Thank you, Mr. Chairman. Good afternoon, ladies and gentlemen. Today, I will introduce new concept about glioblastoma origin cell and molecular signal of subventricular zone in glioblastoma patient. Professor Lee and I proved that the genesis of IDH wild-type GBM is not caused by a somatic mutation in the tumor site, but begins in tumor-free subventricular zone in which the neural stem cell is present. Looking at the right picture, you can see that shared mutations were observed in tumor-free subventricular zone and tumor. According to our Nature paper, it was revealed that starting with mutation of TERT to neural stem cell in astrocyte ribbon area in subventricular zone by aging process, when driver mutation occurs, this mutated neural stem cell migrate to hemisphere and makes GBM tumor. Metaphorically, we refer to this GBM genesis as the firework pattern of cancer genesis. As you know, cancer stem cell refers to a cell that is isolated from tumor and has the characteristics of a stem cell and at the same time has the characteristics of cancer cell. When orthotopic transplantation is performed, tumorigenic capability can be checked as shown here. GBM origin cells are mutated neural stem cells which were isolated from subventricular zone, and these cells migrate to hemisphere and become tumor. It is not known whether this GBM origin cell has neural stem cell-like stemness and neuroglial differentiation capabilities. Also, it is not known whether this GBM origin cell has tumorigenesis ability during orthotopic implantation such as cancer cells. We mixed CRISPR-Cas9 system and Clanok system. We made one plasmid having sgRNA targeting p53 and p10 and Cas9 protein and also CRE. sgRNA and Cas9 made gene deletion of p53 and p10 and also CRE made EGFR variant 3 and reported TD tomato activation from the mouse genome. These mutated neural stem cells in subventricular zone start to migrate and finally made tumor formation. In this genome-edited model, GBM cancer cells were isolated from tumor. GBM origin cells were isolated from subventricular zone. Controlled neural stem cells are neural stem cells isolated from the contralateral subventricular zone and neural stem cell with LAG-G insertion. From now on, I will tell you about the characteristics of GBM origin cells. First, GBM origin cells' sphere formation capability is higher than that of GBM cancer cell or controlled neural stem cell. This is the result of immunocytochemistry and RNA sequencing. Secondly, neural stem cell-related gene expression is increased. For example, in the case of SOX2, it is higher in GBM origin cell and GBM cancer cell than in controlled neural stem cell. In the case of CD133, it is higher in GBM origin cell and GBM cancer cell than in the controlled neural stem cell. In addition, oligo2 was elevated in all neural stem cells regardless of mutation, and NEUN was characterized by increased expression in GBM origin cell. Thirdly, GBM origin cell has neuroglial differentiation potential as shown in this picture. In summarizing the result so far, it can be said that GBM origin cell has a stemness similar to that of neural stem cell. When we examined whether tumorigenesis occurred by implanting GBM origin cell, GBM cancer cell, and controlled NSC into the mouse brain, GBM cancer cell formed a huge mass around 7 weeks, and GBM origin cells survived until 24 weeks and did not form tumors as shown here. This means that if GBM origin cells was originally in subventricular zone, it was destined to migrate and form a tumor in distant hemisphere. But if GBM origin cell was artificially transferred to distant hemisphere, it would lose tumorigenesis capability. From now on, I will tell you the result of RNA sequencing comparing the tumor-free subventricular zone of glioblastoma with cortex subventricular zone of other disease and glioblastoma tumor. After the comparison of human tissues, I will talk about the comparison between the corresponding comparative cells. This slide is a comparison between cortex and tumor-free subventricular zone in GBM. Of course, neurotransmitter and synapse-related signals are more common in cortex, and in GBM subventricular zone, there are more immune-related signals than cortex. The mouse gene was converted to human gene ontology, and compared to control neurosphere, GBM origin cells has a signal related to metabolism and cellular stimuli. Comparing subventricular zone from other diseases with subventricular zone from glioblastoma, there is not much differences compared to other comparison conditions. For example, the dopamine-related signal is higher in GBM subventricular zone than in other disease subventricular zone. Considering that the comparison conditions of previous human tissues are converted into cells, compared to controlled neural stem cell, GBM origin cells has a lot of translation-related and immune-related signals, and brain development-related signals are also increased. When comparing GBM tumor with GBM subventricular zone, signals related to axonal growth inhibition and Notch1 in subventricular zone are elevated. So far, GBM subventricular zone has been compared to tissues in various anatomical locations. The important point is that we need to do more research to find out what's really important in this informatics data. Comparing with cells compared to the previous result, GBM origin cells has a higher translation-related ribosomal signal than GBM cancer cell. Comparison between cells should also be accompanied by functional studies on meaningful signals, as in comparison between previous human tissue results. We have reported in previous study that when making somatic mutations in subventricular zone, these mutated cells migrate more than LOX-C control. When we compare the invasiveness of the cells isolated in genome editing model, it was observed that the migration ability of GBM origin cells was superior to that of controlled neural stem cell, GBM cancer cell, and GL261 cell line. When comparing the EMT-related gene set, it can be seen that Notch1 is raised based on the total expression level in GBM origin cells. In summary, we isolated the cells corresponding to the GBM cell of origin, which is destined to migrate from subventricular zone and become a tumor, and named it GBM origin cell. GBM origin cells, a mutated neural stem cell, has characteristics similar to those of neural stem cell, but when it is artificially transplanted to another site, it has been confirmed that it does not have tumorigenesis ability or characteristics of cancer cell. In addition, I mentioned the signal of human GBM subventricular tissues and the signal of cells. The important thing is that GBM origin cell is destined to become a tumor, but when it is artificially moved, it has been found that it does not have the ability to produce tumor, which means that there is another cancer tunnel mechanisms. Continuous in-depth research on this is essential. I'm really appreciating to my many co-investigators for helping me a lot. Thank you all the sincere researchers in my lab, and thank you to Yonsei University Brain Tumor Section staffs for unlimited support to me. In particular, I would like to thank Professor Jong-Ho Lee and Joo-Ho Lee giving me a lot of advice on this research. I am a vice chair of Scientific Committee, Organizing Committee of WF-NOS 2021 Seoul meeting. Due to the pandemic of COVID-19, the Organizing Committee delayed the WF-NOS 2021 meeting to March 2022 for 1 year. I think WF-NOS meeting is more appropriate to meet face-to-face than online. I hope many people will attend. See you at Dynamic Korea. Thank you very much for your attention.
Thank you, Professor Kang. We certainly know that tumor environment is extremely important in whether it's management or tumor growth. And your data certainly proves. But I must say, and congratulate you for the work, because anyone who has worked with brain tumor stem cells, I worked with Erwin Van Meer at Emory, and it's really difficult to maintain these stem cells and the time for the intratropic model and then doing this work. So congratulations to your team, and thank you very much for a very nice presentation. Please, Rida, next speaker.
Our next speaker is Dr. David Ashley from the USA, speaking on brain immunology and immunotherapy in brain tumors. Dr. Ashley is the recipient of the title Rory David Deutsch Professor of Neuro-Oncology at Duke University's Preston Robert Tisch Brain Tumor Center and is the director of the Preston Robert Tisch Brain Tumor Center. Dr. Ashley's primary research focus is laboratory-based investigation of the role of immunotherapy as a novel approach to the treatment of tumors of the central nervous system. Furthermore, Dr. Ashley is also involved in the design and application of a variety of clinical research protocols in the treatment of children with malignant brain tumors.
Thank you, and for the kind introduction, and also for inviting me. This is a great seminar, as our previous speakers have identified. I'm going to share my screen and hope that you can see it. So can everyone see that slide? Great, perfect. So in the time I have available, what I hope to do is to take you through a very brief journey, touching on some points related to immunology and immunotherapy in brain tumours. So this is a change in paradigm from previous treatments that we've obviously given our patients, and I think it offers an opportunity for us to perhaps change some outcomes. So what I'm going to take you through is some of what I think are the prerequisites for successful immune therapy, and particularly touch on some of the issues that face us in the field in glioma. I'll pause on the tumour microenvironment and discuss some of the genetic and epigenetic drivers that relate between the tumour and the microenvironment. And then I hope to give you just a touch on a couple of examples of early successes that I think we've had that point to the potential of future success. So in general, in cancer immunotherapy, there's a number of prerequisites for success. One is that you can identify targets, and people discuss tumour mutational burden, MHC, antigens, and so forth. In addition to that, you need an orchestrated and effective effector response. And this is reflected in T-cell gene expression profiles, tumour microenvironment markers, and other biomarkers. And then thirdly, there is growing evidence of intrinsic tumour cell resistance to immune killing, and one must overcome this if we're to have a successful outcome. Immunotherapy in tumours really, in simple terms, relates to two parts of the immune system, the first being the innate and the second being the adaptive or targeted response. The innate or more primitive response initiates the immune response to a tumour, and then a sustained response requires an adaptive or T-cell response. The issue being that if there is dysfunction of the innate system, one has a cold tumour, and it requires an alteration that allows the innate system to detect and respond to the tumour and for the tumour to become hot, and then the adaptive immune system can kick in with recognition and response. The problem being, if you have a cold tumour, there's a failure of recognition of the adaptive system and a failure of response. And there's a variety of reasons why this can happen, and I won't spend too long on this other than to say that there are barriers in cellular responses and trafficking, there's lymphopenia, and there's a variety of strategies that can attempt to overcome these barriers using therapeutics such as TLR agonists, oncolytic viruses, immunotoxins in the innate-based system, and strategies that try and amplify the adaptive system, checkpoint inhibition, Treg depletion, vaccine and antigen targeting, etc. But what I'd like to really focus on is that a fundamental aspect of this is if one does not have an adequate innate sensing and response system, there will inevitably be a failure of the adaptive response. And so a cold tumour is not an adequate situation for immunotherapy. And unfortunately, when we look to GBM, GBM is an incredibly cold tumour, and this was a paper published several years ago now that took a large-scale approach to looking at gene expression profiles and tumour mutational burden as two biomarkers of response. And what you can see down the very bottom is that the majority of glioblastoma has neither a high tumour mutational burden or a gene expression profile reflecting tumour microenvironment that will allow for adequate response to any type of immunotherapy. So there has to be fundamental changes. And this is just some data that reflects, for example, poor targets. One aspect of poor targets in glioblastoma, you can see that in the immunotherapy responsive tumours such as melanoma or colorectal cancer, there's a relatively high mutational burden when you compare that to glioblastoma. And in fact, the paediatric brain tumour setting is even more dire if one considers tumour mutational burden as a single issue. Glioblastoma has a very poorly orchestrated effector response and a low T-cell number and inflammatory milieu. So this is just an example of a melanoma on the left of your screen that's been stained with immunofluorescence for T-cells, and on the right-hand side an example of the paucity of T-cell response within glioblastoma. So what about the issue of immune privilege in the brain? And I think over the decades, our concepts around immune privilege have changed. 20 years ago, we felt that immune privilege would be a complete barrier to any type of immunotherapy in the brain. But now we understand that, in fact, immune responses in the brain are nuanced rather than entirely privileged. And it is a major issue in brain tumours, but it's something that we can potentially overcome. So certainly immune environment and responses differ, and there's multiple reasons for that, including differences in Treg numbers and function, microglia, suppressive macrophages, differences in cytokines, and exhaustion by checkpoint blockade, just to name a few. This is some work that one of our team at Duke published several years ago. This is Peter Fecchi's work that actually showed that one of the reasons that patients with glioblastoma and other brain tumours, in fact, have low T cell numbers, is that there's sequestration of the T cells in the bone marrow in the setting of a brain tumour. And this is driven by alterations in a cell surface protein known as S1P1 in patients with brain tumours. And this situation can be recapitulated in mouse models. And Peter's actively working on developing small molecules that for drugs that will overcome the lymphopenia that we see in patients with brain tumours. The reason for the alterations in S1P1 expression on T cells is unknown, and that's another active area of investigation. But what we do know in brain tumours is that there's a number of both epigenetic and genetic drivers of changes in the tumour microenvironment. And here is an example of one. We've heard a lot about IDH mutated tumours today, and the formulation of 2HG. 2HG is an epigenetic regulator, and in fact the downstream effects of that are to reduce the number of chemokine and cytokines produced that would usually encourage T cell infiltration in the brain tumour and an immune response. However, this does not occur regularly in IDH mutated oligodendroglioma in particular. However, one can reprogram this with inhibitors of IDH, and a number of groups now have shown that you can enhance immune responses through manipulation of 2HG. Our own group's been working on the genetic relationship between ATRX mutations and immune vulnerabilities. And in fact, the heat map you can see there differentiates astrocytomas from oligodendrogliomas in terms of the inflamed gene expression profile, oligodendrogliomas being cold, and astrocytomas having much more active immune infiltrates, mainly based upon bone marrow-derived macrophages. Our work has demonstrated that this does relate to ATRX mutations and is driven by alterations in innate-based signalling through a series of toll-like receptors that are important in inducing macrophage infiltration. And in fact, we've been able to model this in immunocompetent mice and can show that there is a survival advantage based upon inflammatory infiltrates in ATRX mutant tumours. I think the tumour microenvironment, we're going to start to be able to really dissect, and Vidya Chandra Mohan and Simon Gregory in our group have been working hard at both single cell sequencing of immune subsets in brain tumours, and more recently we've moved to spatial transcriptomics, which is technology that allows you to do single cell sequencing in 3 dimensions in a tumour. And I think by looking at these two technologies that are complementary, we will start to dissect out some of the aspects of this genotype-phenotype interaction between the tumour and the microenvironment. Again, some of our own work has shown that GBM may in fact have tumour cell intrinsic resistance to immune-based therapies that we'll need to overcome. And this is work we're doing on the STING pathway, showing that a number of glioblastomas have totally absent STING expression, and that upon stimulation are unable to produce an interferon response, which prevents an innate immune signalling within the tumour. And in fact, part of this may simply be driven by large copy loss. This is the area of 9p loss that people famously know is associated with CDKN2A or P16. And in fact, what is often under-recognised is that this same region in glioblastoma carries the genes for the majority of interferons, and that there is both at a genetic and at a RNA transcriptional level loss of many of the interferon genes in up to 60% of glioblastoma. So despite all that, I'm going to finish by just giving you some vignettes of potential immunotherapy that are showing early signals, and I think gives us hope that we can use these methodologies in the future. This is work that's been performed by John Sampson's group at Duke, and John has been able to show that by targeting viral antigens within glioblastoma with a variety of different vaccines, that one can see a very robust survival signal. These are human studies that were published several years ago, and work is ongoing. And in fact, in more recent human studies that we've been doing, we've been able to change the format of the vaccine to peptides, and have shown very robust immune responses in patients in the setting of newly diagnosed GBM, both with CMV peptides, PP65, and also the IDH peptide vaccination in our hands. Looks like it develops nice, robust T-cell responses. So these studies will move into the next phase of efficacy, and we're very hopeful that they'll translate to meaningful responses as well. Several years ago, we published our results of long-term survival in using the poliovirus vector as a method of stimulating innate immune responses and changing the microenvironment. And obviously this is very encouraging. And most recently, what we've been able to demonstrate is that there is a number of patients who seem to have ultra-low tumour mutational burden that's associated with an inflamed tumour microenvironment at recurrence after conventional therapy. And these are the patients that seem to be responding to these oncolytic viral therapies. And so this forms a biomarker. It's predictive rather than just prognostic, and will be able to help us identify responders to immunotherapy in the setting of recurrent glioblastoma. So I'll finish up. We've spoken about multiple barriers that we do need to overcome in terms of target orchestration and intrinsic tumour cell resistance. I think we need to be able to recapitulate the gene expression profile with immune responses in some patients, and I think in part we may be doing this with the oncolytic viral therapy, and there may be other neoadjuvant approaches such as anti-PD-1 in the setting of recurrence that may in part be doing this and targeting similar patients. Certainly low tumour mutational burden is a biomarker for response, and actually we've gone on to show it's also a biomarker for response to PD-1, anti-PD-1 therapy in the setting of recurrence. And this is no doubt immunologically driven. And we've discussed that innate responses in immunoepigenetics may be a key entry point for us in brain tumours to alter the tumour microenvironment with combination therapies. So I'd like to thank you all for your time and the invitation to present our work. Thanks again.
Thank you so much, Dr. David Ashley. Very nice talk. Certainly immunotherapy in brain tumors is challenging, and what you have shown is that there's still hope when we can turn these cold tumors to hot tumors, whether it is virology that can bring that back along. We will go on to our next speaker. With that, please.
Our next speaker is Dr. Sengsai Yeo from Singapore, speaking on mixed reality, augmented plus virtual reality in neuro-oncology, use of Microsoft HoloLens 2 in brain tumor surgery. Dr. Yeo is the head of neurosurgery at National University Hospital, Singapore, and the medical director of the Singapore Gamma Knife Center. He is the recipient of numerous research grants and has published widely in the neurosurgical literature about stereotactic and neurosurgery, neuro-oncology, head injury, and virtual reality neurosurgery. He recently also organized the Asia Pacific Low-Grade Glioma Network.
Yeah, thank you for the invitation to speak. Yeah, right at the outset, I'd like to thank my senior resident, Dr. Swati Jain, who's right with me. So without her, I couldn't have put this talk together. Yeah, so I'd like to thank her right at the outset. Yeah. And secondly, I'd like to thank Professor Arthur Inam. Yeah, I think we invited him to Singapore last year. So I think I congratulate all of you for a fantastic 3-day neuro-oncology meeting. Yeah, I think it's really a world-class meeting, and I learned so much from all the great speakers that have been speaking the last 3 days. It's always better to have a real meeting, I guess, rather than the virtual meeting. So it would have been nice if I could come over to Pakistan. I think the last time I was there was probably, I think, in the early 2000s, when the late Professor Ali Raja, I remember, organized a neurosurgery meeting in Lahore, and that was really beautiful. But I've not been back to Pakistan since. So hopefully, maybe next year we would be able to come to Pakistan. Yeah. I think I've also noted at this meeting that there's a lot of link to Henry Ford Hospital. Yeah, I think one of my mentors in Singapore when I was a resident was Dr. Balaji Sadasivan, who also trained in Henry Ford Hospital. So when I was a young trainee and he'd just come back from US, so he'd mentioned all the giants that he had trained with, like James Alzman and Gauss Malik and things like that. So to some degree, some of what he learned in Henry Ford also diffused down to me. So it's great that that sort of influence really can diffuse throughout the neurosurgical world. And I think also I met Professor Jack Rock recently, the last few years in Myanmar. I think he runs the neurosurgery boot camp there. And we go up from Singapore to help run that too. So it's great that Henry Ford has an influence there. I think he's doing really great work. Yeah, he even took the trouble to learn the Burmese language, which I have not even attempted to do. So I think, Jack, that's really great on you. Yeah. Okay, so my talk today is on something that we have done quite recently, and I'm happy to share this with you. So it's on mixed reality, which is augmented and virtual reality, and its use in brain tumor surgery. Next slide, please. So just to give you some background, I think intraop neuronavigation is probably part and parcel of everyday neurosurgery in most hospitals in the world, including third world hospitals. I guess cost is still a big issue, but I've been to many third world countries, and I've noticed that most hospitals do, not all, but most hospitals, even in third world countries, find that neuronavigation is quite essential. Yeah. But there are some disadvantages to the current neuronavigation. It's still a 2D projection of images. You constantly got to change your focus between the surgical field, and you got to look on the neuronavigation screen. And the instruments that are pre-registered with the neuronavigation can only be used. So recently, I think with mixed reality technology, this has attempted to overcome the disadvantages of the neuronavigation systems. And this allows the surgeon to superimpose a 3D rendered image onto the patient, right? And I think thus far, I think at least with Microsoft, they've largely used it for education, yeah. And some studies have assessed its accuracy, but I think we are one of the first groups to use the Microsoft system for neurosurgery. So just to give you some background, I think virtual reality has been around since the 1990s, and then subsequently augmented reality maybe in the early 2000s, and currently mixed reality is really virtual reality plus augmented reality. So this is the definition of mixed reality from Wikipedia, right? So mixed reality is the merging of real and virtual worlds to produce new environments and visualizations where physical and digital objects coexist and interact in real time. And mixed reality does not exclusively take place in either the physical or virtual world, but is a hybrid of reality and virtual reality, encompassing both augmented reality and augmented reality via immersive technology. So actually, we've been in this business for quite a while in Singapore. Yeah, I think this is a paper we wrote way back in the year 2000. Yeah, which was 20 years ago. Yeah, so I do date myself here. So this is the system we developed in Singapore called the dextroscope, and the acronym Vivian stands for virtual intracranial navigation. Yeah. And this is my colleague, Dr. Ralph Cockrell. Yeah, sort of. Basically, this was mainly a surgical rehearsal tool. Yeah, in 3D stereo, so virtual reality. But its use was just confined to maybe preoperative planning. Right, we couldn't really bring it into the operating room. So I think it was particularly useful for like skull-based surgery, where every tumor is a little different. So this was a commercial product, and I think quite a few hospitals in the world bought this system from us. I think the notable ones would be the late Professor Panetsky in Mainz, who was a great fan of this, and that system still works there. I think Johns Hopkins bought one, Kyoto University, but unfortunately we didn't sell enough. So unfortunately this product died. So that's the reality of the commercial, where you could develop something really sexy and beautiful, but if it doesn't sell, then you go bust. But anyway, we also thought that just using this tool for surgical rehearsal wasn't going to really be a winner. So we actually developed another version. We called it DexRay. So this is one of the first users of augmented reality. So basically, you can think of it. We gave the product the name of DexRay. So you can think of it as a pointer, just like your navigation systems, but there was a camera at the tip of the pointer. Next. So this is, we did use it in some cases, and this was reported in neurosurgery in 2009, more than 10 years ago. So you could actually see through, right? So it's a lot more intuitive than even any of the regular navigation systems available. But we didn't manage to commercialize it. So this was basically just a proof of concept and used on a couple of patients and published. So I think this review in World Neurosurgery is very recent. It's from Duke. So I think it's reviewed by Cho et al. So I think it shows that maybe about 26 cases is kind of small, but these are the officially reported cases of neurosurgery where virtual reality or mixed reality was used. So we worked with Microsoft with the first generation HoloLens, but that was really kind of heavy. So not really very comfortable to use, but there have been reports, especially one from Amsterdam that reported quite a large series of patients. So Microsoft has discontinued this first generation, and this HoloLens 2.0 is the next generation product from Microsoft. And we're fortunate in Singapore that they have an office right downtown, so we work closely with the engineers to develop this product. Yeah. So it's a portable system and with an enhanced holographic processing unit and very easy MR rendering, and we have evaluated its use in a few patients, which we wish to share with you in today's talk. So this is what it looks like, and here's my resident Swati wearing it. Yeah. So this is more than just, let's say, something to view images through. It's actually like wearing a computer on your head, actually. So literally, you probably don't need to even have call up MR images on the computer screen anymore. This can all be flashed directly into your viewing piece, looking through this HoloLens 2. And you can call up anything on the internet too, and you can view it directly. So I'll just illustrate with two cases. So the first case is an extra-axial tumor, right? So this is a 72-year-old female. Yeah, I think we first operated on her more than 10 years ago, and she had a large occipital meningioma. And we had a total resection then, but the histology was atypical meningioma, and 10 years later, she comes back with recurrence, right? And so I think we did offer a gamma knife, but I've listened to Professor Osama El-Smefti's lectures lately, and I think most of these meningiomas have probably changed my philosophy and probably would want to remove them rather than give radiation or radiosurgery. And anyway, for atypical meningiomas, I think the results of a gamma knife are not fantastic. So we used our regular Medtronic stealth neuronavigation as well, just for corroboration. Yeah. And we had this 3D image rendered using the HoloLens 2, and this was then projected onto the patient to assess its accuracy. So this is what it looks like. So you can actually literally see through the patient's head, so to speak, and it doesn't matter which direction you can change your head and so on, and it will still be stereotactically accurate. So we'll show you a video here. Yeah. So you can literally see down to the tumor. Yeah. So of course we're using our stealth here just to check the accuracy, but of course the vision in the future is that you know, it will be so accurate that we don't need to use stealth to check anymore. Yeah. Okay, so some of the learning points in this case, you know, we still had to use surface matching to match the 3D image of the patient in front of us. Yeah, I think, of course, the accuracy of the localization will depend on the surface matching. And sometimes if you have a loss of surface marking after draping, then it requires a repositioning of the image. I think the Microsoft engineers are working on some algorithms to overcome this. So hopefully in the next few weeks, we don't have to do this manual surface matching anymore, and it will be all automatic. The good thing is that this new HoloLens 2, it's really light compared to the first version, and really you don't have much surgical fatigue, even if you wear it for a long period of time. Okay, now we come to its use in intraaxial tumors and using connectomics here. So this is a 56-year-old man who had a 1-month history of expressive dysphasia with right-sided weakness. And the MRI scan showed this very large solid cystic lesion. And he had a systemic workup which showed the possibility of lung cancer. Right? So in view of the large size, we elected to surgically remove this. And we also had DT, being an intraaxial tumor, so we also had DTI images, which were quite preoperative, and we processed them with this omniscient software. This is a software developed by Michael Shugru and his group in Australia, right? Yeah. So this is what some of the DTI images look like. It's not colored yet because in this, this is like the first case for us with Michael. So, but you can see essentially this tumor involves almost all the networks, EFOF, SLF, the central executive network, and the corticospinal tract, all very close by. So I think this is the first time we're trying to incorporate tractography into this HoloLens system. Yeah, so it's very much along the lines of Professor Dufault, where we try to achieve a maximum resection while preserving neurological outcomes. So yes, we do pre-op tractography, intra-op mapping, and awake surgeries at the same time. So this software was created by Michael Shugru and his team. It's machine-based learning, and the important thing is it's subject-specific, and so it's really unique to this patient. So it's a subject-specific version Connectomics Atlas, which is based on diffusion tractography, structural connectivity. So we'll show you some pictures of this. Yeah. So this is what he's developed. You can see the sensory motor tracts there, the central executive network, the language tracts. You can see SLF, EFOF, visual tracts. So I mean, it's really complicated. This I don't think anyone has developed an atlas as sort of comprehensive as this one. So it just shows you the enormous amount of connectivity between different parts of the brain. And right now, I think our vision is to get this all downloaded into the HoloLens so we can actually see these tracks during our surgery. So this is our patient. Yeah, so you can see, unfortunately, we haven't, Michael is trying to get it all color coded for us. So in this version, we just see everything in white. But you can see there's an area right behind here where we went in where there's no activity. So that gives you guidance on where to go so you don't disrupt any of these vital networks or these hubs. Okay, this is a video here. So basically, you know, you can literally just see right through, yeah, without the need to look at the screen, right? So we think this really is the future of neurosurgery. So the aim, of course, is to do it in navigation and just wear this and carry on with the surgery, right? So I think hopefully we'll get color tracks the next time, just so that you'll have more beautiful pictures to look at, you know. And I think it's mixed reality does provide an exciting avenue to explore. It's used in neuro-oncological surgery. And I think right now we've used it for pre-op planning and real-time visualization of these tracks. And this is just preliminary work. And so hopefully our future work, a lot of work required to make this a real commercial product. So in conclusion, I think that mixed reality may eventually replace navigation in the neurosurgical OR. I think mixed reality gives the ability to directly visualize anatomy and connectomics superimposed onto a patient's anatomy. And I think the good thing is cost. You know, it looks very fancy and futuristic and high tech, but the reality, it's actually kind of cheap, you know, compared to the navigation systems available, which usually cost more than 100 grand in Singapore or US dollars. I think this HoloLens 2 doesn't cost more than, I think, 2,000 or 3,000 Singapore dollars. So it's not more than 2,000 US dollars, you know. So hopefully this is a disruptive game changer and can be used in hopefully many or every neurosurgical unit in the world. So that's our hope, that we'll develop something that's useful for global neurosurgery. Thank you.
Thank you very much, Professor Yeo. I think your talk kind of came at almost towards the end of the symposium. Yesterday we had a history by Professor Rosenblum, where he was talking about a needle, and they were putting that needle like in a haystack to find tumors and how far we have come. And I can see Dr. Nam, you know, jumping back and forth, looking at all these new gadgets, and must be thinking what he's going to get for his Christmas gift. So I'm sure he must be excited, but it's amazing to see how far we have come, you know, with technology and how much we can do for the patient and reduce and minimize the discomfort and improve the outcome by all these techniques. Thank you very much. Yes, Shraddha.
Our next speaker is Dr. Andrew Sloan from the USA, speaking on a phase 1 study genetically engineered stem cells targeting MGMT in newly diagnosed MGMT unmethylated glioblastoma, evidence of survival benefit. Dr. Sloan is a board-certified neurosurgeon, physician scientist, and educated at University Hospitals Cleveland Medical Center and UH Seidman's Cancer Center. He is also the director of the Brain Tumor and Neuro-Oncology Center at UH Seidman Center and the Neurological Institute. He also serves as professor and vice chair of neurosurgery at Case Western Reserve University School of Medicine and holds the Peter D. Crystal Endowed Chair in Neurosurgery at UH. He has been recognized by his peers as one of the best doctors in America since 2003 and as one of the top surgeons in America since 2007. In 2014, he was elected president of the Ohio State Neurosurgical Society.
Well, thank you very much, Dr. Inam, and thank you to the organizers for inviting us to share our work. I must say this is really exciting that you guys have been able to put together such an exciting symposium. So I'm going to share my screen with you now. I hope you can all see it. And I'm going to tell you about a phase 1 study that we recently completed and an ongoing phase 2 study that we are just starting. Here are my conflicts. I think we can all agree that treatment for GBM is insufficient and there's a lot of room for improvement. And you can see here a paper we're all aware of, Roger Stoup's study with the ERTC in New England Journal 15 years ago now, where he demonstrated that treating with concurrent radiochemotherapy not only improved survival, but changed the shape of the curve so that there is a small number of long-term survivors. But in the same edition of the New England Journal, Monica Hagee and her group associated with Dr. Stoup showed that the predominant benefit accrued to those patients with methylated MGMT. These folks had nearly a 2-year median survival. Unfortunately, this is less than half the patients. The majority of the patients, the ones who had unmethylated MGMT promoter, roughly 55 to 60% of those patients, got barely a 9/10 of a year benefit, and yet had all the increased toxicity that this concurrent radiochemotherapy was also associated with. So this begs the question, can we inhibit it, MGMT? Now, the answer actually is yes. Duke Group and a number of other groups demonstrated quite successfully that the MGMT inhibitor 06-benzylguanine reproducibly depletes MGMT quite effectively and maximized tumor response and survival benefit in early phase clinical trials with associated significantly improved survival in the patients who tolerate the regimen. But therein lies a rub. It is a highly toxic regimen associated with unacceptable lymphopenia, neutropenia, thrombocytopenia, with high morbidity and mortality. This is likely due to low MGMT expression in hematopoietic progenitor cells, and this approach basically had been abandoned. MGMT, of course, is the molecule that deacetylates the cross-links in DNA induced by alkylating chemotherapy, such as temozolomide. And 06-benzylguanine, as you can see here, irreversibly acetylates MGMT, making it completely inactive, and that's an irreversible reaction. Oops. I am somehow missing a slide. I don't know how that happened. The director of our cancer center, Stan Gerson, made an MGMT P140K mutant. And the thing that's unique about this mutant, and I don't know what happened to the slide, is it has baseline deacetylation activity. So it works just as well at deacetylating crosslinks, but it does not, it's got steric hindrance. It's got a little protuberance here. It does not bind 06-benzylguanine, and so is not affected by benzoguanine. And so our goal was to see if we could dissociate the anti-tumor benefit of combining benzoguanine and temozolomide, which was obviously something that was desirable, with the hematologic toxicity on the bone marrow, which is obviously undesirable. And the concept was that we would try to differentially expose benzoguanine to MGMT in both compartments at a differential basis. In other words, the tumor would not, the tumor would have a high benzoguanine exposure, whereas the bone marrow would be protected. And our hypothesis was that if we could take autologous hematopoietic progenitor cells, genetically engineer them to express this mutant vector, they would then home preferentially to the bone marrow, thus maximizing the tolerance of this combined and somewhat toxic chemotherapy to augment tolerance with minimal toxicity. Conversely, the tumor would not be protected. So we performed a phase 1 clinical trial to test this hypothesis. So this was a trial combining standard of care, which obviously is resection and chemoradiation. And then we used a lentiviral vector carrying this MGMT P140K mutant so as to avoid insertional mutagenesis, which had been previously observed using previous generations of retroviral vectors. And of course, our entry criteria were those were newly diagnosed GBM, IDH1 wild type, MGMT unmethylated patients. These are the worst patients, worst GBM patients, and those who had KPS of greater than 60. The idea here is even if we could transfect only a small subpopulation of these hematopoietic progenitor cells, with each round of temozolomide chemotherapy, you end up killing off some of the intrinsic bone marrow cells and repopulating with these cells with mutant MGMT, such that by the 5th or 6th round, you have, 5th or 6th cycle, you have predominantly the MGMT mutant cells. Our primary objective was to demonstrate safety and feasibility. The secondary objectives was to evaluate clinical response and in vivo enrichments of these P140K mutant cells, and to determine whether these patients who receive this aggressive treatment tolerate this combination of benzoguanine and temozolomide with intra-patient dose escalation without myelosuppression. In terms of safety, I think it was fairly well tolerated. Of the first 10 patients, 8 patients completed at least 1 cycle. 5 patients completed 5 to 6 cycles. Median number of cycles completed was 4. Toxicity was moderate, and hematologic toxicity predominated with minimal non-hematologic toxicity. Here's the hematologic toxicity and the non-hematologic toxicity. 20% of the patients did require dose reduction, but no mortality was observed. In terms of survival, the median progression-free survival was 22 months. Median overall survival was about 2.5 years. But again, remember, these are the worst performers, IDH1 wild type, MGMT unmethylated. This is a small phase 1 trial, so we don't really have a good control group, but we did look at the RPA predicted survival in these patients, both individually and a group. And what we found was we had about a 3.5-fold increase of true survival compared to predicted survival. And actually, we have one patient who's still alive, someone after alive and actually tumor-free more than 5 years out. We saw a number of radiologic responses. You can see that here. And in terms of chemo selection, we found that with each cycle of chemotherapy, the expression of the mutant increased, and then it fell after the chemotherapy, suggesting that some selection pressure was required to drive the expression of the transgene. As predicted, myelosuppression was quite moderate, and both platelets and lymphocytes were fairly stable despite dose escalation with each successive round of chemotherapy. Conversely, in the stem cell population, the bone marrow, the population, the mutant fell with chemotherapy as these cells repopulated into the periphery. But after the selection pressure of the chemotherapy ended, the expression level in the bone marrow increased. We looked at the cells afterwards and found that the viral insertion sites was quite variable, suggesting a lack of clonal dominance, which hopefully will help us avoid insertional mutagenesis, which was something that was previously observed in previous generations utilizing this strategy with retroviral vectors. So in summary, chemotherapy-induced myelosuppression was moderate. 8 patients tolerated at least 1 cycle. The median number of cycles tolerated was 4, and this was, again, increasing doses of benzoguanine and temozolomide with each cycle. 50% of the patients completed 5 to 6 cycles of dose escalation. The median overall survival was about 2.5 years, which was 3.5-fold higher than RPA predicted survival. Gene markings increased by 3 to 26-fold in peripheral blood in response to this chemotherapy, and persisted for somewhere between 200 and 600 days. And finally, no clonal dominance was observed. Thus, genetically engineered hematoprogenitor cells used for this trial were tolerable, safe, and enabled temozolomide and benzoguanine dose escalation, and was associated with increased survival in this small phase 1 study. I'm happy to report that recently the NCA funded our U01 to support expanding this to a phase 2 clinical trial with the NCI. Of course, I have a number of collaborators that I've listed here, and in fact, there's still more, so my apologies for those I couldn't fit in a single screen, and I'm happy to take any questions.
Thank you so much, Professor Stone, for a very exciting talk. I think your talk is a perfect example for all the residents and our attendees who are listening and watching us drug development is one of the major components of any therapeutic change that we eventually help us in taking care of our patients. We're taking care of questions at the end of the talk. I think Dr. Atha would like to add something before the next talk.
Yes, thank you very much, David. Andy, thank you for your talk. And Andy's connection to Henry Ford is that he was at Wayne State. So, Andy, we're just trying to connect people to Henry Ford. So I'll just share a screen here for Noreen to come and say something. Okay, there we go. All right, Noreen, you have 10 seconds. Is Noreen here?
Oh, okay. Yes, Dr. Athar. Thank you so much, Dr. Athar. It gives me great pleasure to announce Pakistan's first Pediatric Neuro-Oncology Symposium to be scheduled on 27th and 28th November. Please save the date. It will be at AKU, and it will be virtual and physical. So our society for event is. It will be a great opportunity for pediatric or oncologists, histopathologists to join from the expertise of experts worldwide in the field of neuro-oncology. Please do save the date and do register. It's open for registration. We are also taking abstracts. I hope this will help strengthen our dream for capacity building of pediatric and adult neuro-oncology across Pakistan. Thank you so much.
Thank you, Noreen. Noreen is 20 years younger to me, but she's 20 years ahead of me. Noreen, I'm following your footsteps with the past now. Okay, back to Rida and Adnan. Thank you.
Final speaker for this session is Dr. Mansoor Saleh from Kenya, speaking on when a headache is more than just an ache, the oncologic approach and management of a brain mass. Dr. Saleh has tenured Professor of Medicine and Pathology and Director of the first in-human early drug development program at the University of Alabama Comprehensive Cancer Center in Birmingham, Alabama. His area of research and clinical focus is targeted therapy of cancer. He recently joined the Aga Khan University in Nairobi, Kenya as the founding chair, Department of Hematology, Oncology, and founding director of AKU Cancer Center. Thank you.
Thank you, Rita. Thank you, Dr. Athanam, and thank you, Adnan, for inviting me. You know, I share the legacy of Dr. Athanam because I trained at Henry Ford, so I will take all the accolades for the good work being done at Henry Ford. I went to obviously Alabama to do my residence and my fellowship and my rest of my training. And Alabama has a very strong neuro-oncology program. Bert Neighbors has a very strong neuro-oncology program. Jim Markert, a very good neurosurgeon. So I really didn't do very much in terms of brain tumors. So I learned more from this than I will be able to share. But I think this topic, I think, is probably very relevant. So let me talk about when a headache is more than just an ache. So if you look at a headache, you have primary headaches on the left-hand side, and then the secondary on the right-hand side. This is, my handwriting is not very good, but it gives you a sense that you have migraine headaches, very intense, usually unifocal. Intense, they come in a wave, stay there for a couple of days. People are photophobic, etc. So they really are very quiescent at that time. Tension headaches are probably you're very frontal, a tight belt around your head, something that probably Athar Enam has had for the past couple of weeks, preparing for this symposium. Cluster clearly are very intense, high intensity, thundering like those hammerjacks on the road. And those are typical thunderclaps that we are always very concerned about. And sinus headaches are usually sinusitis here, including frontal. And hormonal headaches, most women will know them during menstrual cycles. And then you have secondary headaches, talk about SNOOPs. We'll talk about this in a minute. So if you look at tension headaches, a majority of patients will have a tension headache sometime during the lifetime, 2/3 of the patients. Migraines, about a quarter of our patients will have suffered from migraines, routine migraines that go away with time, require medications. Cluster headaches, less than 1%, very intense, not very common, but they're really, really serious when you have them. And then secondary headaches are about 1%. And those 1% are the ones we really need to pay attention to. And I'll talk about that in a minute. So if you look at it, migraine headaches, about 2/3 of patients will have it in frontal. About 1/3 will be more global headaches. Gradual onset, crescendo. Patients really refer to be in the dark, very quiet. You don't want to be with your kids at that time because every motion, every sight will really make them worse. They have photophobia, nausea, vomiting, etc. Tension headaches, things that all of us experience, very tightness around your head, about half an hour, go away most of the time. You can continue to work. Cluster headaches are one of most concerns, where really it's a very intense thunderclap-like intensity. But importantly though, patients who have a headache who meet these criteria may not require additional imaging. What we heard yesterday from Kamel, we heard about from Aisha, Yasser's wife, that headaches really are very important. So if there's a new onset headache, change in character, or headaches that cannot be explained, I believe the clinician needs to give that individual a second thought. And that's going to be my focus of my talk. So, those kinds of headaches associated with systematic symptoms, neurologic symptoms associated with the headaches, new onset, never been there before, and obviously associated with new features that were not there before, and very progressive. When you hear these kinds of symptoms from a patient, do pay attention because they may be different than the usual headaches you experience. So, what about patient-reported first symptoms as a primary brain tumor? What are the symptoms that a patient will present as signs of a brain tumor? Headaches. 1 in 4 patients with a primary brain tumor will complain of headaches. Kamal talked about it yesterday, and so did Aisha when she talked about Yasser's complaints. Generalized seizures, less common, but 1 in 5 patients will have a seizure as part of their presenting symptoms. Clearly a concern. And then unilateral weakness, neurologic focal deficits, or maybe more generalized deficits. And the rest of the symptoms you know about, expressive aphasia, confusion, problem-solving issues, all these things, if they do occur in the context of a headache, that patient requires additional intervention. Do not write it off. So, the brain mass associated with a headache, the causality are basically brain invasion, compression of adjacent structures, and increased intracranial pressure. I remember when I was at Henry Ford, the first thing I had to buy was an ophthalmoscope, an otoscope and an ophthalmoscope. Look at everybody's retina, put that in your HNT examination. I must admit to you, in the last 20 years, I've not seen any resident walk around with an ophthalmoscope. We depend upon the MRI, the CT scan. I think we need to get to the basics, and we were talking about what should be done in the LMIC. In the absence of a CT scan and MRI, retinal examination is clearly very important because 1/3 of the patients will have increased pressure that could be really viewed in some form through the retinal examination. But those are the causal reasons for why patients have headaches when they have brain tumors. So evaluation, we talked about it. It requires a very careful history, neurologic examination, and MRI with contrast. Unless imaging features are atypical, additional testing is often not necessary. And what are those atypical features, i.e., patient may be immunocompromised. You might think patients have primary lymphoma, CNS lymphoma. Uti talked about it yesterday about primary CNS lymphomas. Those are really a collection of patients, a very typical MRI picture, and you might have leptomeningeal involvement. When you have that, obviously you do the LP, otherwise an LP is not required for everybody. What about glucocorticoids? Patients present with severe headaches, neurologic symptoms. Often we believe that there might be a primary CNS issue involved. MRI was positive maybe. We start them on glucocorticoids. Not a problem, but if you do have primary CNS lymphoma, it may really alter the biology of the disease, at least the histology of the disease, and there may be some confusion about the histology. So I would suggest that if you are suspecting primary CNS lymphoma, as suggested by Huti, wait for your corticosteroids until you get a biopsy. And if you do give corticosteroids, the dosages are usually the standard 10 milligrams IV or PO at the onset, and then 4 milligrams every 4 hours or 8 milligrams twice a day, just to control the headaches, control the neurologic symptoms, to give you time for the patients to at least be quiescent to be able to do the rest of the workup. So imaging, CT scan, very helpful in the ER, can be done in 10-15 minutes, all done. The MRI really takes a lot longer. So most of us will do a CT scan. We'll pick up a hemorrhage, we'll pick up edema, mass defect is easily recognizable with midline shift, hydrocephalus, or herniation. All those can be picked up very easily by your first test, which is the CT scan. The MRI obviously is very mandatory. The sequences you require are DWI, the flare, the contrast, very critical when we talk about brain tumors. And sometimes performed, and we've heard about this from our speakers, MR perfusion, spectroscopy, as well as specific molecular imaging, much of which we cannot do in the LMIC. But those are the features of what we look for in an MRI. So let's talk about, you got the MRI. The MRI was done. If you see a dural-based tumor, the likelihood that this is a meningioma is very high. I borrowed this slide from UpToDate, and certainly meningioma would be one of the things you worry about if you have a dural-based mass. However, if it is not dural-based, one of the concerns you might have is, for me, the oncologist, rule out metastatic disease. What makes you think about metastatic disease? Well-circumscribed mass, multiple lesions, always a concern for metastatic disease, more than one compartment involvement, and a known history of cancer. So you've got to worry about metastatic disease. If you have any of those concerns about metastatic disease, yes, then you look for the primary site, lung, breast, melanoma. The patient may already have a history of metastatic cancer or cancer per se. That makes it much easier for you. So if you do have a history of cancer, on the left-hand side, you can see that a lot of these things need to be done. Most of the time, you may not need a biopsy. The patient is active, systemic disease, no biopsy required. If the patient is NED, no evidence of disease, after 3 years of primary surgery or treatment for a prior cancer, I think it's very important to get a biopsy because this could be a secondary malignancy. So that is the workup for metastatic disease. But what if there's no suspicion for metastatic disease? No suspicion. As Huti talked about, you really need to consider primary CNS lymphoma, homogeneous enhancement, periventricular, near the corpus callosum lesion, and prominent diffusion restriction. When you get those signs on the MRI, you really worry about primary CNS lymphoma, and that may sway your treatment in quite a different way. So as Dr. Athar Enam talked about yesterday, every mass does not to be sent to the neurosurgeon and to the radiation oncologist. There is much to be done between then and now because of the evaluation we're talking about. So if you think about primary CNS lymphoma, if it's yes, avoid glucocorticoids. Those are the patients who need an LP and flow cytometry and really looking at immunoglobulin gene rearrangements in the CSF, because that will be very useful for primary CNS lymphoma. On the other hand, if you do not think this is a primary CNS lymphoma, this has been the subject of today's and yesterday's and day before yesterday's symposium. Primary brain tumors, and I will not go about them, but the talks that we've heard is neurosurgery, pathology, as well as chemotherapy and radiation, all are given. So that is basically for the residents and fellows, what to do if a patient has an MRI and how to guide yourself as you go below to find out the actual diagnosis and the treatment. With a differential diagnosis of a brain mass, we talked about primary brain tumors, as you've heard about. Metastatic disease, 1 in 2 patients who have a brain mass, at least in the US, will have metastatic disease. So the primary CNS tumors are much lower than that. So you really have to consider, although the focus of the symposium has been on primary CNS tumors. Vascular abnormalities need to be considered, hemorrhage, stroke, etc. Infarcts, posterior leukoencephalopathy syndrome has to be considered. And obviously, in countries like yours, in Pakistan, like mine, in Africa now, TB, malaria, HIV, very, very important. So certainly, not rush to the surgeon, not rush to the radiation therapist. You need to really think through about the systemic involvement that may be involved in your particular context. But the question is, brain metastasis is a first manifestation of advanced cancer. Is it possible that a primary lesion in the brain may be the first sign of brain metastasis, not from a primary CNS tumor, but for metastatic cancer, solid tumors? And here's the answer from this particular publication from about 2 years ago. Retrospective analysis, 2,500 patients who had a primary brain lesion as a first symptom of onset to clinical presentation. About 1 in 5 to 1 in 6 had bone marrow metastasis as their first symptom of advanced cancer. So very important that if you do have a lesion, it may be solitary, yet 1 in 6 patients might have that as a primary source of their malignancy that needs to be determined. As you can see, a majority of them had the diagnosis of metastatic cancer or at least solid tumors within the first 3 months, and a few of them had no evidence of cancer, yet this was not a primary CNS tumor, it was carcinoma of unknown primary. The treatment of choice for brain metastasis for that particular patient is very unique. And I won't dwell into it, but you have to then treat the cup, which will be once you get the diagnosis, as well as treat the brain metastasis. So if you do have brain lesions as a site of first presentation, what may be the primary lesion? Where may the primary lesion be? Majority of patients will have lung cancer. Second will be breast cancer. Third will be melanoma. Renal cancer number 4, and colorectal cancer number 5. So if you look at solid tumors that metastasize to the brain, and the brain is a first sign of disease, I think lung, breast melanoma has to come through your diagnosis, and the treatment of those lesions can be quite different. It's very important to have that in mind. So if you look at that particular publication that I talked about, this one that said 1 in 6 patients have a primary site in the brain, but their actual disease is systemic cancer. The outcome basically is the same in most people. Lung cancer, median survival will be about 3 to 6 months. Other cancers will be 7 months. But once you have brain metastases, the median survival in general is going to be in terms of months, not in terms of years. On the other hand, if you have patients who have a known diagnosis of cancer within 3 months, their median survival is going to be about 7 to 8 months. With patients with CUP, their median survival is half as much. So it's better to get a diagnosis of the primary tumor because the outcome will be quite different. This slide is very important. If you identify a cancer which has the ability to target therapy, median survival is 20 months, compared to those for whom no targeted therapy is available, median survival is approximately about 7 months. And we'll talk about those 3 diseases. Lung cancer, breast cancer, melanoma, all have targeted opportunities. So you can see that if you all have a primary lesion in the brain, and those are the 3 sites of systemic disease, you are better off because your median survival will be 20 months. On the other hand, all the others, renal cancer, yes, we might have a targeted therapy, but for others, we may not have targeted therapy and the outcome is going to be very poor. So in adolescents and young adults, primary brain tumor are more common than metastatic disease. And as we've talked about the last couple of days, low-grade gliomas are most common. Adults, 30 to 40, metastatic cancer is going to be the most common condition and not the primary low-grade oligodendrogliomas. But glioblastomas, obviously, will be the most common malignant invasive brain tumors in patients who are older. But if you look at it overall, and I think one of our speakers just talked about it, Tracy talked about it, almost patients. Obviously non-invasive, locally advanced, not the typical invasive cancer we talked about in terms of primary brain cancer. On the other hand, the rest will follow through. So it's important to realize that meningiomas may be really one of the first things you mentioned. And as you saw in my flow diagram, it is going to be durally associated, a very typical clinical picture, will require a biopsy, but certainly that is the most common brain tumor you will see in patients with brain masses. So I don't look at MRIs very frequently. I let the radiologist tell me about it, but I have to think about this as I prepare myself. T2 imaging, obviously, fat is bright, water is dark, ventricles here. New blood is bright, a brain bleed will be bright. Useful for anatomic detail, very helpful for vascular changes, which require contrast, and very helpful for disruption of the blood-brain barrier for metastatic disease, which obviously requires contrast. This T2, T1. T2 is the opposite. Fat is dark, which is here. Water is bright, here. And flow is dark when you have blood vessels. very useful for anatomic detail, especially for CSF spaces. Most lesions will show very quickly on T2 imaging. However, T2 cannot distinguish between lesions from the CSF, especially if you're next to the ventricle. And just to show you, here's T1, just the positioning T2. Fluid is black. Fluid is white. Fat here is going to be bright. So clearly a differentiation between T2 and T1 images. Importantly, though, however, is the flare. Flare will recognize free-flowing water, CSF.
Hello, Dr. Saleh. We're over 15 minutes, so could you just please wrap it up.
I think we've lost his voice, have we?
You have my voice?
Now
Right
we
now.
Okay, sorry. What about when you have metastatic disease, neurosurgery versus whole brain RT versus SRS? Single brain lesions, obviously the size, the mass effect, symptoms will be very important in what to determine. And the question is, what do you do? To assess the efficacy of surgical resection for brain metastasis, randomize patients with a single MET to either surgical resection plus RT or RT alone. It is clear surgical resection plus RT is superior to RT alone, both in terms of survival as well as time to recurrence. So the conclusion is, for single brain metastasis, patients will benefit from surgery plus radiotherapy much better than radiotherapy alone. What if you have multiple lesions? Here's a study done with multiple lesions. Clearly, brain RT, whole brain RT is better for local recurrence. However, if you have multiple lesions, is stereotactic radiosurgery post-surgery better than overall? And you can see that patients who've had SRS after resection do better than patients who had surgery alone. So certainly you need surgery, you need RT following that. SRS is as good as whole brain RT in terms of local control. The overall outcome survival is the same because you can salvage patients who will progress. So the overall outcome is surgery important.
Dr. Saleh, I'm sorry to interrupt, but we're running a little late right now.
Dr. Saleh, your lecture is actually very interesting and great lecture. We want to have a special session with your lecture for our trainees. It's
Yes.
really about basics.
So the randomized trial will show that whole brain RT is beneficial, but SR is clearly much important. But finally, and I think something that Nan would like to know more about it, for patients who have targeted therapy options, lung cancer, you have a number of options here that are beneficial that could obviate radiation and obviate surgery. For breast cancer, we have Herceptin, lapatinib, and for melanoma, you have a wide variety of BRAF, anti-MEK, as well as anti-PD-L1 options. So if you have the three cancers, lung, breast, melanoma, you should think about systemic therapy first, as opposed to jumping to the neurosurgeon or the radiation therapist. So in summary, every headache is not a CNS tumor, but 1 in 4 patients will have a CNS tumor. Diagnosis is very critical. And my last slide is this: do not neglect every headache. Precision medicine will be a game changer. But the question is, will our patients, the LMIC, have access to this revolution? And the question is how? It does take a village, as Coleman and Aisha have talked about it. And success is never given, yet we have to give it our all. So I thank you. Sorry for running over.
No, thank you so much, Dr. Saleh. Whatever you are talking about is the most important thing for our residents and most of the practicing physicians out there. It's just that we are already 20 minutes beyond time. And I would like all the speakers, if they can come up. And we'll probably begin from, as we say, from bench to bedside. We have had, very interesting sessions all the way from basic science to clinical practice. So I'll start with Professor Kang. First of all, thank you for inviting us for the symposium, and we'll be looking forward to it. You showed us that the stem cells, when you once take them out, they really don't behave. So where do you go from there in terms of your lab's future work and showing the stem cells being different? Professor Kang?
Is he on? I don't think so.
I don't see him.
No, I don't have it. Yes.
All right. So we'll move on. You know, Professor Zadeh, thank you so much. You know, it's always very important in a low middle income country where, you know, we are always questioning precision medicine is the way to go. And as physicians, when we go to all these...
Sorry, go ahead.
As physicians, when we go to these meetings and, you know, when we come back and we say, what did we learn? And can we implement it in a low middle income country like ours? You know, 15% clinical impact is huge. We talk about absolute benefit in adjuvant chemotherapy of 3 to 5%. You know, being the next president of SNO, I don't want to put you on the spot, but how do I take this, you know, good data and use it? And what can we do as a LMIC moving forward to bring this thing on? And that's just the tip of the iceberg. You know, for the last 2 days, you've been with us and you've seen that especially neuro-oncology is becoming so molecular and everything. Tracy Batchelor just talked about precision medicine, all the, you know, small molecule inhibitors. I would like to hear your comments.
Yeah, thank you, Dr. Jabbar. Maybe I can go first and then pass it on to Tracy. I think it's a challenge, as you said. How do you uniformly implement something that's new across different centers and institutions, regardless of where in the world we're at? One point I would make about the methylation, however, is that it is relatively straightforward. You really need 5 unstained cuts of a paraffin section that could be shipped to labs that do methylation. There's few labs, but they're accessible, and the results can be made available to you. Sometimes faster than perhaps getting histopathology diagnosis, because you'd have to send those sections to be stained, etc., to some of our trained neuropathologists exists. And so I think that data can actually be generated for methylation in a relatively straightforward manner. And I mentioned that we already do provide that service for centers outside of Canada and Canada as well, but across globally, where people, and we get patients actually contacting us now more to ask whether they can get the methylation testing done at our sites. And I know that more and more centers across the world are picking up on using this technology, but it's certainly something that's relatively accessible. And in fact, for places where there's no dedicated trained neuropathologist, you could argue that methylation can help with the diagnosis to be made. And I agree with you, 15% is a significant difference in clinical management for patients. Of course, it's 15% of your challenging cases, not all comers, but nevertheless, it's a significant percentage. I think for some of the other molecular tests, methylation can give you the standard IDH, 1P19Q, MGMT, etc. For some of the other specific molecular tests that you need for targeted therapy, you may need to actually then rely on next-gen sequencing panels. That again, I think is accessible with being able to send the samples across centers. And one more global approach may be with all the societies in neuro-oncology that are being established in the world, that one of the common cross-cutting tasks that we can have as the various societies is to identify the centers that can actually serve as reference centers to send some of these samples to, and identify centers across the world that have capabilities of doing these tests so that you can refer to them in a more ready fashion and create a map of the world where these centers exist. Of course, being sensitive to not creating sensitive competition for these centers, and that's always generated with the notion of being able to get resources or revenue from having sent the test sent. You know, in Canada, we're a little bit perhaps fortunate. We don't use it as a revenue source. And so it takes away the potential of creating a sense of competition. But I think one thing that we can focus on, and perhaps SNO can lead this, is to create a map of the world where main centers that can do next-gen sequencing and methylation to act as referral centers for the rest of the world would be the first step. Tracy, I don't know if you have anything to add to that. And it's great to see you, actually.
Nice to see you via Pakistan. All well said. I don't have really much to add at all. I'll just say a word or two about some of the drugs that are now, I mentioned earlier, these are very niche indications right now that the approvals are for. The NTRK inhibitor, the MEK inhibitor, and the mTOR inhibitor. These are also drugs, the way that it works in the US is that once the patents expire, there are generic forms that become available. And everolimus has been around a long time, so I don't know for sure, but I would imagine that patent has expired. So there will be cheaper alternatives to these drugs in the US, at certain time points. But I think even outside, there are companies that will or do manufacture some of these drugs and make them available. So that would be another way to access them. So and then I think there are going to be others coming. I think I didn't have a lot of time to go into it, but selumetinib is also very active in pilocytic astrocytomas in kids that are BRAF altered, about a 30-35% response rate that appears to be durable. It's also active in certain other pediatric low-grade astrocytomas. There's a paper in Lancet Oncology last year. So I think we're going to see a flurry of these, which is great for our field, but a flurry of these that will be coming to the forefront and will likely get some approvals in the next few years. And then in terms of the testing, everything Gelore said I agree with. Some of these are, some of the targets are identifiable by immunohistochemistry, which is also straightforward and can be done locally, such as, the BRAF V600E point mutation, IDH1 mutations. Most of these are picked up with immunohistochemistry. So there are some ways around, the sequencing. And then Gelore is right. I mean, we all do, we're contacted all the time from colleagues around the world, and we will do testing here once, with tissue that's sent here and provide that feedback. So there are a number of ways to get around it. No one easy solution though to a big challenge that you correctly identified.
Thank you very much. Point well taken. I think, since it's also capacity building, it's capacity building on the histopathology side and the lab, commercial laboratory side. We can send patients, and I think, the idea of mapping all those centers who can do it for us, because our pediatric neuro-oncology really moved forward very fast since they aligned themselves with, with Canada. The program with Eric Buffet has really helped it move forward, not only in terms of management, but also with the diagnosis and other things. So I think that will be really important. There's a question that came in twice before I miss it. I just want to ask by Gelore. Professor Saad, the question is that, did you do global DNA methylation, or those were targeted genes that you tested?
No, it's a global methylation. So either the first version of the Illumina array was 450,000, and the newer one is 850,000, 850K. So it's global methylation, and you look at the signatures and patterns that reflect each tumor type.
So coming back to Tracy, which patient to test and when do we test? One of the things it's very unique to Pakistan is that some of these, IBs are within patent and they are available from the gray market. You rightly said everolimus has been there for such a long time, but even newer drugs, second generation, third generation drugs are available out of patent. And yes, they are not, and that's a fact. We are managing patients, they are not available when you go formulary, but they are available. Patients are getting treated and they are benefiting. The example is sorafenib, which is coming from India, and it's 10 times cheaper than the actual sorafenib. So when we write a prescription and a patient goes out and buys it because everything is out of pocket, if he or she takes that medicine tomorrow, I don't know what he took. So for us, maybe access is not that of a problem, but getting them tested for these is a challenge because nobody's going to do them commercially because they ask me how many patients you're going to send me. And that's where we kind of get stuck. We've been trying to get BRAF, KRAS, NRAS, BRCA, and these are the tests where you have numbers. Talking about the ones that we just talked about for mTOR, for MAC inhibitors, for NTRK, that's one of the challenges. So I'll be interested in hearing, and it's open for the panelists, in your experience, and you've been working with low middle income countries, something that you can tell us, know what we can do in our capacity to make things better.
I'm not sure that I have an answer that could solve that problem or challenge. I think you started the question with which patient should be tested, and I think right now, the reality is that we really don't have any targeted drugs that work in glioblastoma, which is the most common primary malignant brain tumor. Of course, we do test them because we have the ability to do it, and we're trying to enroll patients on clinical trials. But beyond immunohistochemistry for IDH1, perhaps MGMT, I'm not sure how much more you would need to do for practical management of a glioblastoma patient right now. It's where you get into these subtypes where it becomes, the possibilities of these alterations would go up. For example, glioneuronal tumors, again, a rare glioma subtype, but a third of them do have NTRK fusions and a third of them have BRAF mutations, BRAF point mutations. So I would, if I was thinking about practice, I would tailor, my testing to the populations where there are some known alterations and there's some known available options. And I think, we covered some of those, and these are, again, rather niche right now, but I do think it will expand. These areas will expand. And, one of the examples that I gave in my talk was craniopharyngioma, which is, there's now an NCI-sponsored study, which might lead to the drug, a BRAF-MEK combination being approved. So I think you'll see, incrementally, you'll see more and more of these roll out in the future, which will kind of widen the spectrum of where you want to think about, tailoring, customizing these tests. But for the more common, glioblastoma right now, I'm not sure beyond what we discussed, there's more to do there. But and I'm not, we talked about immunohistochemistry, we talked about the availability of academic partners to do some of these tests. Of course, there are commercial vendors who will do these tests, these panels, these onco panels, but again, those are, those tend to be expensive.
Thank you. So say, for instance, you have a patient and you're doing them because of clinical trials. If there is no clinical trial and you're finding a new mutation, do you have a trial where you are putting, depending upon whatever mutation you get, you're using a targeted agent, or it's only after?
There are a few trials like that. So we've made the argument over the years that we should open up our our phase 1 programs in the US to primary brain tumors, because historically a brain tumor has been excluded from phase 1 trials for reasons that are not altogether sound. So we are seeing more of these sort of tissue or disease agnostic trials that enable you to enroll, for example, a glioblastoma patient who may have a rare genetic mutation. The example that I would use is that from the talk is the entrectinib. The patient who was treated with entrectinib, that patient received it on a clinical trial that was sponsored by the NCI that allowed any solid tumor with an NTRK fusion to enroll. There are not a lot of those, though. Hopefully, we'll see more, but there are a few of those where you can enroll based on the alteration and not the histology. Now, you can use these drugs off-label in the US if they're approved. And have we done that? Of course we have. So, there are rare types of gliomas that have BRAF mutations, PXAs, for example, pleomorphic xanthoastrocytomas, have a pretty high frequency of BRAF mutations, and we've certainly used some of the BRAF-MEK combinations off trial for those patients who have recurrent or progressive disease. We like to do it on trial because we want to learn something and contribute to knowledge, but we also want to appropriately manage and offer best therapy to our individual patients. So occasionally we'll use some of those drugs off trial.
I had a question for Dr. Ashley, but I think he had to leave for another meeting, and maybe one of the other panelists may take. In fact, I can ask Professor Sloan to address it. We heard of a really nice talk about brain immunology, and we saw data about brain tumors being cold. We have learned a lot when we, one of the things about when you come back to LMIC, you treat everything. When you are in US, either you are a neuro-oncologist, you are just a lung cancer doctor, or so on and so forth. And we have seen that in head and neck, they are converting cold tumors to hot tumors by giving radiation and then giving immunotherapy. Something like that for brain tumors? Your thoughts?
I'm sorry, is this for Dr. Ashley?
Well, he left, so I was wondering either you or anyone else can address.
Sure, I'd be happy to try to address that. So actually there's considerable literature that, that you can combine radiosurgery with checkpoint inhibitors. This is particularly true in melanoma, and there's new data also in lung cancer. So radiotherapy seems to be potentiating anti-tumor immunity. Whether I don't think there's good data that converts that it makes these tumors hot in a molecular fashion. In other words, I don't think there's molecular evidence for them being hot, but it seems to be efficacious. There are also suggestion that if you look at Duke's work, particularly with the poliovirus vaccine, the data is quite impressive for recurrent GBM, that these viral vectors, and we can talk about the potential mechanisms. It's not exactly clear what happens, but it seems to induce a response against an antigenic response against CD155 in the tumors, which sort of creates a bystander response that then educates the immune system to attack the recurrent tumor. Again, I don't think there's a huge amount of molecular evidence that these tumors are, quote, hot, but we seem to be seeing a response to these tumors, which we presume is immune-based, because it works often not only at the site of the tumor that we see as enhancing on MRI, but also non-enhancing tumors that we see, that's hyperintense on FLAIR or T2 weighted imaging, which presumably also has tumor, often then develop inflammatory response. It sometimes looks like recurrence, but then many times it has gone away, suggesting there's an immune response there. I don't think we can prove exactly that that's happening, but I think that is what most people think is happening.
Thank you very much. Professor Yo, thank you so much for your talk. When is it commercially available for our surgeons?
I think we're just working on it with Microsoft as a project. So I don't know, but I hope within 2-year time frame, you know, if that works, then I think we can roll it out, and I think it would be good for neurosurgeons even in developing countries, because you don't have to invest huge sums in, you know, either Medtronic Stealth or a brain lab system, because this is like much cheaper.
I can go on, but I think we have gone over time and people have other commitments. So I'm gonna just thank all my speakers for wonderful talk and bearing with us where we go beyond the time. And I would hand over to Dr. Shehzad Shamim for the closing remarks and Dr. Athar for taking over.
Before we do that, Shehzad, before your closing remark, you guys have noticed that there are like all these young faces popping up on the screen. And that's because I wanted all of the students who have been playing an important role in arranging all this thing to come up. And you know, this is my source of energy, these students, really. And the residents too, but then there's no resident over here at this point. So if we can all smile, I want to have this picture as a portrait, and you know, just bring your face closer if you are not close enough to the camera. And Sharif is going to take a screenshot. So yeah, that's good. Dr. Yo, your resident was missing, so
yes.
Yes. All right. Okay. Here, Sharif, let us know when you're done.
Sure, sir. Done, sir. Thank you.
Wonderful.
Thank you.
This is like a virtual group photo, is it?
It is, yes. We have to take, so I've been trying to figure out how to do the clapping in the virtual, you know. So I think we will learn. This is a different way of communication. That is. Dr. Shehzad, please go ahead.
Okay, shall I go on?
Yes, please.
Okay, so it has truly been an honor to be a part of the 1ANOS, or the first annual Neuro-Oncology Symposium. As you know, this symposium was endorsed by major neuro-oncology societies, including Society of Neuro-Oncology, the World Federation of Neuro-Oncological Societies, the European Association of Neuro-Oncology, the Asian Society of Neuro-Oncology, and the Society of Neuro-Oncology of Sub-Saharan Africa. The new normal of web-based meetings, you know, the virtual meetings allowed us to engage such prolific experts from all over the world, and to be able to showcase from a Pakistani platform a meeting which, at least to me, is as good as any international meeting one hopes to attend. It also helped us to keep the cost very low so more people could attend it. I mean, a meeting of this scientific caliber anywhere in the world would cost a fortune for attend. The 3-day meeting comprised of 6 scientific sessions and a special session, and included also the launch of Pakistan Society of Neuro-Oncology, which I'm sure will one day be considered a historic moment. Over these 3 days, we were very fortunate to have over 40 neuro-oncology experts from 5 continents, and included people from all the specialties. So basic scientists, medical oncologists, radiation oncologists, radiologists, surgeons, pathologists, and public health specialists, making it a truly global and inclusive event. However, there's always room for improvement, and surely we will be looking at how best to expand on our experiences gained through this meeting, and your feedbacks will be invaluable. Now, in concluding this event, I must thank a few people. Firstly, the more than 1,200 attendees over the last 3 days. This overwhelming response by itself tells us that PASNO will be pushed hard to organize an even better 2ANOS. Secondly, our speakers. To all those present here, thank you very much. To all those listening to us, or to all those who spoke and are not listening to us, thank you for supporting us despite this being a weekend, the time differences and the usual difficulties associated with web-based interface. I mean, how many times have we heard you're on mute, please unmute yourself? But this is also, I guess, a part of the new normal. I would then thank the NSIG, the Neurosurgery Interest Group, the voluntary group comprising of our medical students. Some of you can be seen here. And these are some of the brightest and the most energetic lot you can find on the face of this earth. So thank you very much, all of you. I will thank our organizing committee chaired by Dr. Athar Enam and co-chaired by Dr. Adnan Jabbar. I will then thank my scientific committee, including my co-chair, Dr. Nolin Mushtaq. And finally, I would thank Sharif Charania, the one-man wonder behind almost all the logistics involved with the meeting. Now, if this was an auditorium, Dr. Athar would have requested a massive round of applause for Sharif, for he truly is a superstar. And finally, and this is, I promise, the last thank you, the man whose vision or whose dream we have been living for the last 3 days, the chair of the organizing committee, the founding president of PASNO, and to be honest, also the de facto chair of the scientific committee, my teacher, my mentor, Professor Athar Enam. Thank you and congratulations, sir. And with this, I conclude this meeting. Thank you and good evening.
Thank you, everyone. Aziz Heather, we missed Aziz. Thank you very much for Aziz to Aziz Heather. Have a good day, good night, good evening, good morning, whatever, and bless you all. And I bid you all peace. Thank you. We will see each other.
Congratulations.
Thanks, Rocky. I think that went well.
Yes.
Congratulations.